We'd like to try to get everyone to move in and take a seat in the auditorium. Those that are outside, if we could encourage them to come in. I'm sorry we're starting a little bit late, and that's because the valet parking was backed up a little bit, and we wanted to give everybody a little extra time. It's an amazing group of people that we have here. This is amazing. The attendance is fabulous, and thanks to everyone for coming. And I think you're going to enjoy what you're going to hear today. I'm Paul Cullis. I'm a neurologist from Detroit at St. John Hospital, and I also happen to be the president of the Michigan Parkinson's Foundation. We're pleased to present the 2017 Symposium in conjunction with the Van Andel Research Institute's Grand Challenges in Parkinson's Disease. As I'm sure most of you are aware, this Grand Challenges is an annual event which has been going on for the last 2 days, and it's about basic science research in Parkinson's disease, and it's really been very interesting and very exciting. Researchers are on the verge of actually discovering what causes Parkinson's disease and what we might be able to do to reverse that process. And this is really very good news and provides a lot of hope for patients because we're on the verge of actually getting to treatments that may actually affect the course of the disease as opposed to just treating symptoms. We'd like to thank the Van Andel Research Institute for hosting our symposium today. Special thanks to Kim Cousineau and to Dr. Patrik Brundin, who you're going to hear from in a minute, for making all of this possible. It's certainly an exciting event. I'd like to thank our sponsors for their financial support. Those include St. Jude Medical, which is now known as Abbott, AbbVie, Acadia, Impax, Medtronic, US World Meds, and our lunch sponsor, Mercy Health Holstein Neuroscience Institute. I would like those of you that are here to visit the sponsors as you're walking by. They have a lot of useful and interesting information for you, and we need to thank them because they've provided financial support. The people that I would really like to thank also are Mary Sue, Kelly, and Diane, who are the people at the Michigan Parkinson's Foundation that make all of this possible. They do all the heavy lifting, and we certainly owe the success of this to them. For those of you that don't know, the Michigan Parkinson's Foundation is located in the suburban Detroit area and serves the entire state of Michigan. It's a very unusual organization in that many Parkinson's organizations are national or serve local areas, but the Michigan Parkinson's Foundation is a very active organization serving the whole of Michigan, and our mission is to help people with Parkinson's disease today. We do that by providing education and support to people with Parkinson's and related disorders, their loved ones and care partners, and the physicians and other allied health professionals who diagnose and treat those affected by the disease. We have ongoing patient education programs throughout the state, and in fact, we just had a similar symposium last week in Iron Mountain in the Upper Peninsula, which was very successful, and we'd like to thank the people that organized that. In addition, we have, believe it or not, 71 support groups throughout the state, financial assistance for medication, financial assistance for daytime respite care, referrals for neurologists and other community resources, caregiver education programs, an annual research grant most recently issued to a professor from MSU Medical School. And for more information about the Parkinson's Foundation, you can always contact the foundation. They're easy to get in touch with. The phone number is on all the brochures. 248-433-1011 is burned into my memory. There are a number of events that have been organized throughout the state to raise resources. Our walkathon in southeastern Michigan was back in May and was extremely successful. There's a walk scheduled for Portage and Kalamazoo on the 21st of October, and I would very much encourage anyone in the area to participate. That will be at Celery Flats Historical Area. And I love if you haven't seen it, the motto for the walks are "Give Your Soul for Parkinson's Disease," as in SOLE, which I think is a very clever slogan. We have also had an event for the last 2 years in which we've had a cruise on the Detroit River on the Ovation yacht, and we just had another one of those, the second annual, organized by Jeff Latham, and that was also very, very successful. So we would encourage you to be involved in the Parkinson's Foundation, to be involved in the fundraising events, and to take advantage of the services that are offered. So it's my pleasure, first of all, to introduce Dr. Patrik Brundin, who I was just telling you about. He will be presenting updates in Parkinson's disease research, a lot of which we've been hearing about in the last couple of days. He's professor and director of the Center for Neurodegenerative Science at the J. Van Andel Endowed Chair in Parkinson's disease. He's published over 325 papers on Parkinson's disease and related topics. He obtained his Ph.D. at Lund University in Sweden in 1988 on intracerebral transplantation in Parkinson's disease. I had the opportunity of being in Lund recently, and those of you that haven't been there should go. It's a fabulous place and a university town, and very pleasant to visit. He's been involved in a lot of research on Parkinson's disease, basic science research, and in 1992 he obtained an MD at Lund University. In 2008, his team described Lewy bodies in grafted dopamine neurons and suggested that a prion-like mechanism operates in Parkinson's disease. He leads the Center for Neurodegenerative Science at the Van Andel Research Institute, and his research team is focused on experimental models and novel therapies in Parkinson's disease. Dr. Brundin is one of the most cited scientists in neurodegenerative disease with a focus on Parkinson's and has received numerous awards for his work. He's the co-editor-in-chief of the Journal of Parkinson's Disease and is a consultant for several biotechnical and pharmaceutical companies. And it is a pleasure to introduce to you today Dr. Brundin on his home turf. Thank you.
Thank you. So first of all, good morning, and I'd like to welcome you to our institute. I'm very happy there are so many people here. We've had 4 very exciting days leading up to this. As you heard, 2 days of grand challenges and something we call Rallying to the Challenge, which is a patient-centered meeting that we have parallel to the scientific meeting, and it's in the room next door. So next year, I urge all of you to come also to Rallying, and you can walk in between the Grand Challenges scientific meeting and the Parkinson patient-centered Rallying to the Challenge. I'm also very happy you're in Grand Rapids today because it's a beautiful day, and you know Art Prize is still going on. So those of you staying a few hours after the end of this meeting should take the opportunity to walk around downtown and see all the art that's on display. When being asked to talk on about this theme, updates in Parkinson's research, I realized there are so many things one can talk about. I've selected 5 topics. One is why are the changes in the nervous system, what are the changes in the nervous system that underlie Parkinson's disease? And then I'm going to spend quite a lot of time discussing, can we predict the disease and can we identify triggers? I will also spend a lot of time on the greatest research challenge, and that is without a doubt to find a therapy that slows progression of the disease. There is no such therapy at the moment. And one approach is to repurpose drugs, and I will spend some time on this because it's something we're working on here in this building together with the Cure Parkinson's Trust in England. And finally, I will speak a little bit about a very exciting development. I hope that in Michigan and in Chicago there will be patients who will undergo brain repair in the next 1 to 2 years from now. So this is a totally new program. I've been preparing this talk for over a month, but I put the finishing touches to it at 1:45 this morning. That's why you don't have handouts. I've added stuff from this week, and I've also added this typo here, which of course it should say, what are the changes in the nervous system? So what are the changes in the nervous system that underlie Parkinson's disease? And in 45 seconds, I can give you a quick overview of what happens in the brain. I think almost all of you have heard of dopamine neurons. These are the black pigmented neurons in the midbrain, right about here in the inside your heads, that produce dopamine. And they progressively die, so that in Parkinson's disease, after 25 years of disease, there's very few of these left. They are the key to all the motor symptoms. In addition, there are these accumulations of misfolded protein or protein aggregates that are called Lewy bodies and Lewy neurites. They were discovered by somebody called Frederick Lewy or Lewy 105 years ago. He didn't know what they contained. Precisely 20 years ago this year, we found out what the main constituent of this is, and this has meant a huge leap forward for the research. The protein is called alpha-synuclein. Initially, these are restricted to a couple of brain regions. I'm going to talk about that later. And then progressively, they spread, these clumps of protein, throughout the brain the longer the disease has been there. And I will explain that in a minute and why that is important. Now, you may wonder, why does this happen? We spend most of our time wondering, why does this happen? It's our primary job. Those of us who do basic science try to figure out the reasons this starts and what are the drivers that pushes this process forward over the years as the disease gets worse. I'm going to show you a series of 4 slides that look a little bit complicated, but the aim is to simplify it into just 4 concepts. If you look at these processes that cause this, we know there are many molecules that are involved, and I just pulled out some from the literature. Here they are. All these molecules seem to be involved in these processes. You may recognize alpha-synuclein, which is the main constituent of those Lewy bodies. But if we look at these in a very critical fashion and consider what they're doing in cells, we can cluster them. So this group of proteins here seems to be involved in the protein clumping events. So somehow they're involved in that process. But there's also another group of proteins that seems to be involved in energy deficiency. This is an important mechanism, and it's so important that the 2 days that we were here discussing science this week, the total focus was on energy metabolism and how that is deficient in Parkinson's. There's a third cluster you can make, and that's free radical stress. I think everybody's heard of free radicals. These are highly reactive molecules that can destroy the constituents of cells. Sometimes you will see that one molecule will appear in 2 clusters because there is overlap. Some things that are involved in energy deficiency are also involved in generating free radicals. Finally, very importantly, and actually the theme of the Grand Challenges meeting in 2013, some of these molecules are clearly involved in inflammation. And I believe you all know what inflammation is. When you get stung by a bee and it's red and swollen and hot, that's inflammation. We can get inflammation in the brain too. And these are some of the molecules involved there. 4 clusters: inflammation, protein clumping, energy deficiency, and free radical stress. So these belong together some way. So the processes interlock in some manner. And our job in the basic science laboratories is to understand how these interact inside cells. Now, some people might argue that there is a clear line of events, a sequence of events. And some of the people who were here in this room the last 2 days would argue energy deficiency is at the top of the pile. That's what happens first. And then the others follow on, because we know that when the energy factories in cells, they're called mitochondria, when they start to fail, we know that they can start producing free radicals. And we know that free radicals can destroy some proteins, including alpha-synuclein, and then you can get clumping. And we know that protein clumps can stimulate inflammation. So this is a plausible idea that it follows this line, but it could equally be that it starts with inflammation, because we know that a virus that all of us have had as a kid in the gut, when you get gastroenteritis, can be caused by viruses. We know that such viruses cause inflammation. And we know that such viruses, this summer we learned that, can cause protein clumping. And that could then cause the energy deficiency and then the free radical stress. So we're not quite sure which order these things happen in, and we have to be open to the possibility that it isn't a linear process, and this is why the research is so hard. It might be a vicious cycle where each one of these 4 mechanisms interlock in a dynamic fashion, so at different phases of the disease, different parts of this process are driving. So that was a very short introduction to something that's actually quite complicated, to be honest, and where we are not fully on top of this. But I can say if I'd given this talk when I was a Ph.D. student in the 1980s in Sweden, the only box here that I would have talked anything about, I think, is this free radical stress. So these other 3 boxes appeared after 1987. The Lewy body was known since 1912, but pretty much ignored. People just said it was there and we didn't know what it contained. So we know a little bit about the mechanisms, but wouldn't it be wonderful if we could predict the disease and actually identify the triggers? Because then we're moving towards the idea that we maybe could prevent the disease. Now, if we're going to predict the disease, we have to look at a very long time course, basically decades. We need to look at the whole life of the individual who eventually, typically at age 60 or so, gets Parkinson's disease. And we have to understand all the events. And what we've learned when looking at this process is, in recent years, that the molecular disease process, those 4 boxes, probably starts quite early in life and gradually gets worse and worse. And the first visit to the doctor probably occurs long after the first one of those 4 boxes lights up. So we have a phase of the disease which precedes the first symptoms. So here, there's no tremor. There isn't any stiffness that's noticeable. Maybe the golf game gets worse, but could get worse for many reasons. And there is no slowness of movement. So a bit of rigidity, perhaps something very, very subtle, but not really noticeable. And we call this phase the prodrome. This is a relatively new concept. It's emerged in the last 10 years, and you probably heard about this. Please, those of you coming now, lots of seats at the front. So come on up. The first 3 rows are almost empty. So we have learned about the prodrome in the most recent decade that the prodrome is characterized by a series of sometimes subtle symptoms, and they are not necessarily related, or typically not related to movement. So there'll be sleep disorders, the loss of sense of smell, constipation, and depression are typical components that we see in the prodrome. My message is we must understand this phase better, because clearly this is where the interesting opportunities are to prevent the disease in the next generation of people who are destined otherwise to develop Parkinson's. So there is a lot of research focused on this prodrome and trying to figure out what's going on during that time. So do you know what the most important early event is in the cause of Parkinson's? Can you imagine? People are pointing to their nose. That's a good answer because some of you might know that I'm very interested in the loss of sense of smell. I'm actually going to take you back even further. The most important early event is the time of conception, because we inherit genes from our mother and father that will lay the foundations for whether we are at a very low risk genetically or a high risk of getting the disease. We think, and I will tell you this in a moment in more detail, that 5 to 10% of Parkinson's disease cases are inherited in a familial way, but the remaining 90 to 95% then are not truly inherited diseases, but genetic risk plays a role. Plays a role so that maybe it underlies a third of the total risk for your disease. So 90% of you here who have Parkinson's don't have a familial form. You may have slight risk factors, and they're going to be very subtle. But what happens sometime early in your life, you had an environmental insult. And maybe you had a second environmental insult in your 20s. These environmental insults we don't fully understand, but it could be things such as viral infections that could affect the nose, could affect the gut, could also be pollutants, and I'm going to mention this in a second. So we want to figure these out, and we think they occur very long before the first symptoms, even before, obviously, the prodrome. By definition, at the time of the prodrome, the disease process has already started. Now, why don't you get the disease immediately here, the first time you have, let's say, if it's a gastroenteritis? Why don't you get Parkinson's disease at age 14 when you had your diarrhea at age 13 and a half? Well, we think that our bodies are set up to defend ourselves against these bad protein clumps and are set up to reduce the inflammation and so on. But at some point in life, those defense mechanisms fail. And we know one thing for sure, that is that normal aging is an important risk factor because the majority of you here are not 14, 15 years old. The typical age of onset is in age 60. And you also know that there are many young people who get this, but in the big picture of things, they are a small minority. So we think that we could be exposed to several environmental insults that could be partial triggers of the disease, superimposed on an inherited genetic risk. And then once normal aging sets in, this whole process just starts going off. Maybe this vicious cycle speeds up. Some of these things here are of particular interest to our institute. Our institute is interested in something called epigenetics, and I will mention in a second what this is. This is something that actually, epi means on top of. It sits on top of the genes. It's something that controls our genes, and it's a complicated thing, but I'll explain it in a second. So if we could predict who is destined to get Parkinson's disease, could you imagine how wonderful that would be in terms of developing therapies? Because then we would have the opportunity to intervene perhaps before the vicious cycle has started to spin. And that is really the aim of a lot of the research today. It doesn't mean, as you will hear in a minute, it doesn't mean we are not interested in curing the disease and people already have it. But we think in the long term, this would be the ideal situation to intervene very, very early. So let me describe now genetic risk for you in some detail. And this is a little bit complicated, and this is something I spent a couple of hours up until 1 this morning trying to make slides that can be understood. And if anybody has questions, so raise your hand, and I'll try to explain it. So I've already said that only 5 to 10% of people with Parkinson's have a truly inherited form. That means we can identify a genetic mutation in a certain gene, and we can even go back and see if it came from the mother or the father. And in some cases, actually you need a mutation both in the mother's copy of the gene and the father's. That's called autosomal recessive. And I also said already that the remainder have some genetic predisposition, but you need probably an environmental insult, and you need normal aging on top of that. So let's look at genetics in an oversimplified way. So the things I'm saying now are not 100% true because it's much more complicated. Things I'm saying now is the sort of stuff I learned in medical school in the 1980s, and I wish it was like that, but it's much more complicated today. So DNA is the code that we have in every cell in the body. It's inside the headquarters of the cell called the nucleus. The DNA is a series of letters in the textbook, and those letters will code for proteins. They're actually little chemical entities that are tied together and form that double helix that I think you've all seen. In fact, a coffee break, look outside in the reception. Over the reception, there's this Chihuly sculpture, which is a DNA helix. And each one of those balls is one of the letters in the DNA code. Artistically, not in reality. So imagine this is the DNA that we have in one human, this long line, and we now know, for the past 15 years, we've known that there are about 20,000 parts of the DNA in every cell, in every body, in our body, except the red blood cells maybe. There are 20,000 genes that code for proteins. The amazing thing is the number of genes that code for proteins in little worms and flies is not too different from this. So we have about the same number of variety of proteins actually throughout the animal kingdom. In about 10 to 15 out of these 20,000 genes, it's known that changes in the gene, which we could then call mutations, that alter the protein actually cause Parkinson's disease. So you can see it's a very, very small number. And some of those genes will, with certainty, cause Parkinson's disease if you carry the mutation. In others, they are just risk factors that increase the risk, so you have about 10% instead of a 1% lifetime risk. Now, here comes the tricky part, and this is super important. So only about 2% of this long genetic code that every cell in the body has is actually genes. This DNA has, these are the genes I've just depicted, 2% of this line. Here are 3 genes that code for proteins, so these are 3 out of the 20,000. The DNA has all this other stuff that a few years ago we used to call it junk DNA. Still sometimes called junk DNA. It meant that we didn't know what it was, it didn't seem to code for anything, and there were all sorts of theories why it had ended up there. And then people started saying, but surely we can't have so much DNA that doesn't do anything. Let's call it the dark matter in the cells. So it does something, so it's dark matter, and we'll have to figure out what it does. And we have actually hired a couple of people in our center that are focused on the dark matter in Parkinson's disease. Because we now understand that the dark matter actually regulates the genes. So the stuff that isn't the actual genes, but the stuff in here, can decide whether you have a lot of that protein or very little of that protein. And we have learned that changes or variations in the code, that's like code, right? In the code between the genes, in this dark matter, variation in here actually decides whether you have a 1.3% risk and you have a 0.8% lifetime risk of getting Parkinson's. So these are very subtle changes, right? But we can discover this. So we're talking then about the genetic predisposition. That's in the dark matter. And these studies are very complicated, and that's why I hesitated talking about them, but I want to share it with you. So this is how many authors you need to do a scientific study on the dark matter. These are the authors here. This paper came out in Nature Genetics 3 years ago. And they had now done a meta-analysis, large-scale, genome-wide association. They were basically looking in the dark matter. And they found 24 places in the dark matter, in all of that code, 24 places there were things that regulated risk. And they had to look at 13,000 people with Parkinson's and 95,000 people without to be able to get the statistics for this. And we were excited about this, and we thought, okay, now we know. Let's go after those 24 places in the dark matter and try to understand them. But you know, science always advances. And a week ago, another paper came out with a lot of authors. In fact, some of these authors are actually International Parkinson's Disease Genomics Consortium, 23andMe research team. You may have actually sent your DNA there. These authors are hundreds of people, each one of them. They're big groups of people. And what they found was another 17 places where there are genetic modifiers, predisposition for disease. And the reason I gave you this long, convoluted introduction is it's good to know what is meant by genetic risk. And if you're talking to your family, and if you are the only person in your whole extended family that has Parkinson's disease, it's not very likely that you have a mutation in a gene that causes Parkinson's, but you may have some of these risk loci. But these are tiny changes, tiny, tiny, and you need hundreds of thousands of individuals to actually discover them. But now we're diving into the dark matter. And Vivienne Labrie is an assistant professor we recruited from Canada a year and a half ago. She's upstairs. Together with her, we are now trying to understand the stuff that's in the dark matter. Which gene is it controlling? Because I said it could regulate the genes. So we're trying to see what it's associated with. And here's a map of some of these dark matter locations. And this is where serendipity comes in. So these are the first 24 places they found changes in dark matter. This is called a Manhattan plot because it looks like Manhattan. The higher the tower, the more important is the genetic variation. But if you look very carefully, there's one thing here called ACMSD, and it's quite high. So this regulates risk for Parkinson's. I'm married to my wife. My wife's name is Lena Brundin. There she is. She also works here. She's a psychiatrist, and her interest is actually suicide and depression. But when she saw this Manhattan plot and this enzyme, she said, wow. Remember, there are 20,000 proteins in the human genome. Her primary interest in life in the last 7-8 years has been that one out of 20,000. But she's been studying it in the context of depression. So she said at breakfast, we've got to do something with this. This is very interesting. ACMSD seems to be involved in depression and in regulating Parkinson's risk. So we started to dive deeper into this together with Vivienne, and we got a grant from the Michael J. Fox Foundation. In 2016, the grant came, and we've just finished working for a year on this project. And what we did is we created a Parkinson's model in rats, and we induced inflammation in the brain in rats, and we destroyed the black dopamine cells. And then we used the protein, the enzyme that my wife is so interested in, ACMSD, and expressed it with gene therapy. So we used a virus to make lots of ACMSD because we think it protects. And maybe you can see, if you use gene therapy for ACMSD, that damage is dramatically reduced. So we're learning that the genetic variability in the dark matter that could regulate ACMSD could actually regulate inflammation. And this is totally novel in this context. So we've counted these cells, and we see this is the ones that have inflammation. They have reduced number of dopamine cells, but when we have gene therapy, it goes almost back to normal. And now we're trying to get the NIH to fund this, so keep your fingers crossed. In the next 3 weeks, they're reviewing our proposal. We realized to really make people believe this is a good idea, we have to do the reverse. What's the reverse? Let's remove the protein and see what happens. So we spent 18 months, just to give you a sense of how long these things take, to remove the protein from mice with genetic engineering. So we had to go into the mouse embryos and cut out the gene with a fancy genetic technique, and then we put the mouse embryos into another mouse, and they were born, and then we suddenly had mice that lacked this protein. Our idea would be what, do you think? If the protein protects, if we remove this protein, what do you think happens? It gets worse, right? That's the idea. And we're just in the middle of this, even though we've been working on it for 18 months. But lo and behold, when we completely knocked out the protein, the number of black cells in the midbrain is reduced compared to here in the wild type, the normal mouse. So we're seeing a reduction in the number of cells. At 2 months of age, it's about a 20% reduction. 2 months is a young mouse. We now want to follow these mice until they get old. Maybe we have created a model of the genetic risk that some in this room, in the whole world actually carry. This is a common genetic risk, and it changes the risk a little bit. So that gives you an idea of how we and the rest of the Parkinson world is working on trying to dig into the mechanisms. We know that that protein is involved in inflammation, so it's one of the 4 boxes, and we know that it is a commonly occurring variation in the population. So let's talk about environment just briefly, because everybody is interested in this notion that environment can cause Parkinson's disease. And for a few years, we've known that there are two places where the disease seems to start, which are in touch with the outside world. It is, of course, the nose, and it is, of course, the gut. Both those parts of our bodies are in direct contact with the outside world. Those parts of our bodies are exposed to viruses, bacteria, fungi, pesticides, pollutants that are coming out from industries. So we're very interested in this. So we just got a grant that just started a month ago from actually the Department of Defense to study airborne pollutants as triggers of Parkinson's disease via the nose, the olfactory system. So how do we do this? Well, once again, we look at mice, and in a small syringe, we make Lewy bodies, those protein clumps. We make them in the laboratory. We suck them up into a syringe, and we micro-inject a tiny, tiny droplet of Lewy bodies into the olfactory bulb. This is the part that the mouse smells with, and we've sectioned it and looked at it in a microscope, and the red stuff here is where we make these injections. Our idea is that something can happen in the nose when you're exposed to pollutants, and that will cause the formation of these Lewy bodies, and then they will spread throughout the brain. And we actually already know that if you inject Lewy bodies in the nose, they do spread. After 1 month, they have reached these areas, and then you can see after 3, 6, and 12 months, more and more of the brain is involved. And this is what you heard in the introduction that I've been interested in since 2008. We think this is prion-like spreading of the bad protein. And this is not unlike what we see in mad cow disease or Creutzfeldt-Jakob or chronic wasting disease in elk. So we're thinking that the initial event could be in the nose in some patients, and we believe, but we haven't tested it yet because the grant just started a month ago, that if you're exposed to airborne pollutants from California, it'll get worse. And the reason that the airborne pollutants are from California is that our collaborator is from University of Southern California. He's a specialist in looking at these airborne pollutants, has been doing it for decades, and we just teamed up when we did this. In addition, in this project, we have an epidemiologist from East Lansing. Honglei Chen just moved there. He is, like all epidemiologists, a person who looks at populations and asks questions like, if you live close to a highway for most of your life, are you more likely to get Parkinson's disease than if you live in the middle of nowhere? We don't know the answer to that quite yet. So those are things we will be pursuing during the next 4 years. And during the latter part of those 4 years, we'll also be looking at ways of interfering with this process. Because if this airborne pollutant causes an inflammation, maybe we can use anti-inflammatory drugs to reduce it. Now, I said the other place where there might be environmental insults are in the gut, and here I want to highlight things that have just started, but they are super duper exciting. And I can only share a little bit because it's not quite ready for prime time. But Vivian has been working since she arrived 1.5 years ago on this wonderful project, together with my wife and a little bit me, where she's been looking at the gut. We know from the literature that the Lewy body-like changes that you can see in the brain in Parkinson's disease occur in the gut. So there are nerves that make your gut move, and you can feel sometimes as the tummy rumbles. Of course, the muscles are moving the gut and the stomach around. That's controlled by nerves, and those nerves can form Lewy body-like changes. We don't know if they can, in a prion-like fashion, move from the gut up to the brain, because that's the idea that if it starts here, those bad guys can move along the nerves up to the brain and cause the disease we call Parkinson's. We don't know that for sure. But we do know that people with Parkinson's are almost always constipated years before they have problems moving. And maybe these changes in the nerves here are the reason that people with Parkinson's get constipated, and then eventually a few of these clumps will sneak by up the nerve here to the brain and spread in a prion-like fashion. And there is reason to believe that might be true, because people in the era before there was good medication against peptic ulcers sometimes had that nerve cut as a surgical therapy. This was a way to cure severe peptic ulcer. And a group from Denmark looked at the lifetime risk of getting Parkinson's after they'd had this surgery. It's called vagotomy. And they saw that the people who had the nerve intact, they actually never had the nerve cut, but they were the same age. They were the controls for the ones who had the nerve cut. The cumulative risk of getting Parkinson's disease, you can see it's twice as high in those with the nerve intact as those who had peptic ulcer and had the nerve cut. So PD risk is reduced if the nerve has been cut. Now, it's obviously not a therapy, because this would have to be done, you can see, up to 30 years before. We don't know who's going to get Parkinson's, so how could we do that? And it's not a surgery that's trivial. But I hope I've given you a sense of that we might be able to predict the disease in the future, and we might know where some of the triggers are. And once we understand them, I hope we can develop therapies, because this is the great research challenge. There are currently no therapies that are absolutely certainly slowing disease progression. As you will hear in this section of my talk, we think we have some in the pipeline, but it is still the holy grail of Parkinson's research. If we can solve this, it'll mean a huge difference for the lives of everyone affected by Parkinson's. Medical research has invested a total of about $23 billion in therapies that they've tried to slow Parkinson's research. All of them have failed. $23 billion. The annual NIH budget is about $32 billion. So it's 2/3 of a year of research for everything. Every disease has gone into this, and it has failed. The equivalent of that. And why is that? Well, the disease process is complex. I hope you've realized that with those 4 bricks that I showed you. But it's also very difficult to test this. So even if we had the ultimate drug, it's not that easy to show. And why is that? Because it's a slow disease. That's one of the reasons. And if you look here, this is the first visit to the doctor where the diagnosis occurs. And here's a measure of some functional capacity. You know, can you walk? Can you turn your hand like this 20 times rapidly? All those things you can test. And we know that over years and decades, this gets worse. Eventually, if nothing is done, and if you live for 30 years, there's a high risk, as we all know, that people with Parkinson's could end up in a nursing home if they don't have adequate support at home. And our idea is that we could start a new therapy that would change that curve. So people would always live at home and be independent. It isn't quite a cure, obviously. You can see that. But just making a nick in the curve here would be amazing. It would mean a lot. But the problem is, to run a drug trial for 20, 30 years is impossible. You can't do that. The cost is unbelievable. And patients wouldn't follow it anyway, because, you know, who would be patient enough to try an experimental drug for 20 years, not knowing if it was working or not? And even worse, who would want to be on the placebo control for 20 years? So what we have to do, and this is the challenge, is we have to do 1-year drug trials or maybe 2 years. So we're looking at this tiny time window, and we have to hope that the drug is so effective that there is a separation. And therefore, we have to have relatively large numbers of patients, and this also becomes expensive. And it's further complicated by the fact that not all people with Parkinson's are the same. They don't always follow exactly the same route. Some will progress slowly, and they will actually always live independently at home. Others, for some reason, will progress more rapidly and be severely ill within 5 to 10 years. What we have understood is that people with Parkinson's are individuals. The notion that it is one disease, that we can throw everybody in the same bucket, isn't really valid. And we're trying to get out of that thinking, and we're trying to create something that's called precision medicine for disease modification in Parkinson's disease. So patients belong to distinct nodes or clusters. We need some kind of biomarker, we need well-defined groups, and that will help us perform better trials. Smaller group numbers, short duration, because they are really the same type of patient. So here's one patient with lots of motor symptoms, here's one with lots of pathology, here's one with a lot of genetic load. And our thinking is that people will each have a little bit of everything here, and they will need different types of pills, different therapies. To do this, to achieve this, researchers, such as us, have to figure out biomarkers. These are things we can measure in the blood or in the spinal fluid or see on brain scans, and we don't have these yet. It is very expensive to develop new drugs. In the central nervous system, it takes 35% longer to develop a drug for the central nervous system than it does for oncology, for example, for cancer. And to create one new drug is now about $5 billion if you include all the failures, and only 8% of the drugs make it. So you have to test 12 drugs all the way through before you get one that works, and in the case of the brain. And if you include then all the cost of the failures, you get up to about 5 billion. So even if we didn't have any failures, if we had the greatest drug today for Parkinson's, from day one to the CVS pharmacy, or Walgreens, because I'm Swedish and he was Swedish, so Walgreens, it's $200 million, even if there were no failures. So, can we do this in a smarter way, in a cheaper way? Could we possibly use an approach called repurposing drugs? What's drug repurposing? Well, that's using a drug that's already available for something else, like diabetes, asthma, or inflammation of some kind, and then trying it in Parkinson's. Because then we know that the drug is safe, it's been in hundreds of thousands of people, we know its mechanism of action usually, and if we know that mechanism of action is relevant to one of those 4 boxes, inflammation, protein clumping, free radical stress. What was the 4th one? Energy deficiency. Thank you for that prompt, because that's the one I'm going to show you right now. It could be much cheaper and faster, and everybody knows the classic example of drug repurposing, I think. There was a drug that was developed for blood pressure. Viagra. The side effect turned out to be more interesting than the initial, and that's a $1 billion drug for Pfizer. So we've been working with the Cure Parkinson's Trust in what's called the Link Clinical Trials Initiative, which is a drug repurposing program. And this was really a priority and almost a brainchild of one of the founders of the Cure Parkinson's Trust, who sadly is no longer with us. Tom Isaacs, on this picture here, when he was visiting us 3 years ago, passed away at the end of May this year. He had had Parkinson's for 22 years, and he was 49 at the time when he died. Suddenly, unexpectedly, very, very sad. He stood on this stage several times, and those of you who met him knew and know that he was an incredible source of inspiration, an absolutely wonderful patient advocate. So, to honor that and to carry on his legacy, we are devoted to pushing this forward even harder. We're looking at all sorts of drugs. This is the first 25 drugs we looked at, and seeing if they're suitable for repurposing. We have an expert committee of about 14 people that sit down for a day and a half and go through all these drugs every year. We have new drugs every year. We've been doing this now for 6 years. And the first year we did it, we prioritized an anti-diabetic drug called Bydureon. It's right at the top here, got the best score. We knew that they had results coming out suggesting that if you change, or if you give the anti-diabetic drug Bydureon, you might change the disease course. The drug is also called exenatide in another formulation. Here are the people who got exenatide. This is how poorly they're doing in their Parkinson's. The higher the score, the worse. When they got the drug, they actually felt a little bit better. 12 months later, they hadn't changed from when they started. People who were not getting the drug progressed and got worse, which is what you would expect with Parkinson's over time. But most importantly, when they stopped giving the drug, followed up the same patients a year later, there was still a difference. So that suggests what? 1 year of an anti-diabetic drug permanently changes the disease course? Does it do something in the brain that protects cells or rewires the brain? So this was sufficiently exciting. It was not a definitive answer because this study wasn't blinded. The controls knew they weren't getting drug. The ones who were getting exenatide knew they were injecting something every day. But this might be the first drug that does this. So together with Cure Parkinson's Trust, this blinded placebo-controlled trial was designed. They had a slightly different formulation, so the injection was only once per week. It was slow release. It was cheaper to make the placebo pen. It was affordable then to do a proper randomized double-blind placebo-controlled trial. This could be a landmark in Parkinson's research and in Parkinson's therapy. Because when this came out in The Lancet on the 3rd of August this year, what it showed was that the people who were on exenatide got remarkably better initially, maybe part of that is placebo after 12 weeks, but then stayed stable for 60 weeks, whereas the placebo people, now they knew, or didn't know that they weren't getting the drug. They could have thought that they were getting the drug. They just got worse over time. And the last period here, actually the treatment had stopped, and the difference is maintained even when the treatment has stopped. This was presented by Tom Fultony. We gave him an award on this stage 2 days ago, the Tom Isaacs Award, because in memory of Tom Isaacs, who had pushed this program so hard, it is wonderful that maybe it is already, through luck, the first drug prioritized is coming to fruition. And we have a bunch of drugs that are at different stages now of trial. This is the one that's published. This one is being tested in Plymouth. You see there are over 200 patients now. This is being done in France. This is being done in London. This will be done in various places across the world. This is being done in Los Angeles. And these are in different stages of planning. This is actually being done in France. This will be done all over the US. And the nice thing about this one, all the work for that one, that's also an anti-diabetic drug. These are different drugs. That's a cough syrup. That's an iron chelating drug. That's a cholesterol drug, etc. But this one, the work was done here together with 2 people from Kalamazoo, Bill McDonnell and Jerry Colker. And we're super excited that that one is actually now being prepared for trials. It's also an anti-diabetic drug that affects the, what did you just say? Energy metabolism. So that's the key. So thank you for your prompt. And we met again this year on the 6th floor here. So this is the group that does the prioritizing. So it's a mixture of experts in neurology and laboratory science, and importantly, several patient advocates are in this group. I would say about a third of these people are patient advocates from the UK, Canada, US, who give their input and their thoughts on the different drugs we're discussing, because it's always safety first. We don't want to give them a dangerous drug. We want to give them safe drugs. What do you think was our top drug this year? Can you guess? If you've been following the news in the last 3 weeks, 4 weeks, did you see this news? Quite a remarkable story. And if I go back one slide, the guy hiding next to me is Clemens Scherzer, who is the professor who did this study at Harvard. He looked at people and animals with Parkinson's that had been given an asthma drug. When he gave the asthma drug, in this case clenbuterol, to a Parkinson animal, you see here are the dopamine neurons. There are not so many left here. When he gave the asthma drug, there were many more left. So the asthma med is protecting them. But even more compelling, when they looked in a Norwegian cohort, because Norway has fantastic medical records, they could see that the probability for not having developed Parkinson's disease in a general population declines like this over years of follow-up. But if you have taken an asthma drug, in that case in Norway it's called salbutamol, if you've taken a lot of asthma drug, your risk is dramatically reduced. If you've taken intermediate, it's slightly less reduced. And if you've taken a low amount of asthma drug, it's still statistically significant, but just a tiny bit of reduction. They looked at 4.5 million people. So maybe this asthma drug, when it goes into trial, hopefully in a year or so from now, will be another part of our arsenal in slowing Parkinson's. We know a little bit about how it might do that from the initial study. Finally, those of you who already have Parkinson's disease, you don't just want to have it slowed, you want to have it reversed. That's the last 2 minutes of my talk. So Jeff Cordova, who's another professor working in the institute here, we've known each other for 30 years. We have both worked with cell transplants for about 30, 35 years. We've teamed up with a company called Cellular Dynamics International. They're owned by a Japanese company, but they're based in Madison, Wisconsin. And they are making stem cells from skin. It is a discovery made by a Japanese scientist called Dr. Yamanaka, and he got the Nobel Prize for this, 2012. It's a huge discovery. He made the discovery in 2006. 6 years later, they give him the Nobel Prize. That almost never happens. And it's a sign of how important that discovery was. So what is the discovery he made? Well, he saw that if you take some skin, just pinch a bit of skin, there are cells called fibroblasts in the skin. They can grow. Then you add 4 genetic factors in the laboratory, and the trick was to identify which 4. He had 20,000 to choose from. He picked the right 4. Didn't do that in a weekend. He had to think a little bit. And then he selected cells that looked different, and they looked like real stem cells. Then he took those and converted them, or he and others now have converted those stem cells into the dopamine cells that you need for Parkinson's, and you can transplant them. So Jeff Cordova, who was on that picture next to me, he's done this. That's Jeff there. And he's just published a paper where he's taken human skin cell-derived stem cells made into dopamine nerve cells, put them in a rat with Parkinson's disease. Here's the transplant, and this is what they look like in high magnification. They make dopamine, they're human, and they actually work. And we know from prior work that if we can make the right sort of dopamine, neuron, dopamine nerve cell, it has a very good chance of reversing the motor symptoms. We're not promising anything else. And we know that it might work for 20 years. So we can't affect the other symptoms with this, but the motor symptoms we can. So we're planning a clinical trial here in Midwest. The patients will be operated at Rush University in Chicago. They will be recruited from Rush, but also we hope to recruit some of them from U of M or even this area possibly, because the brain imaging will be done at U of M. So I find that kind of exciting because this is something I worked with for 35 years, and actually now being able to do this here with the support of someone who can afford to do it, Fujifilm, makes it really stimulating because we spend a lot of time trying to get money to do projects. Here we know they want to do this. We just have to guide the science. So with that, I'm happy to take questions, and I want to thank you for listening.
All right, it's my pleasure next to introduce Dr. Eric Ahlskog. Dr. Ahlskog is an internationally known expert on Parkinson's disease. He's professor of neurology at the Mayo Medical School and former chair of the Mayo Clinical Neurology section of movement disorders in Rochester, Minnesota. He's a full-time clinician with the majority of his Mayo practice devoted to movement disorders and especially Parkinson's disease. He's published 241 manuscripts in peer-reviewed medical and scientific journals, most related to Parkinson's disease and movement disorders. As many of you know, a little over a decade ago, Dr. Ahlskog published a book for people with Parkinson's disease and their families called the Parkinson's Disease Treatment Book. Several variations and updated forms of this are currently available. The new Parkinson's Disease Treatment Book, Parkinson's Disease Treatment Guide for Physicians, and Dementia with Lewy Body and Parkinson's Disease Dementia, Patient, Family, and Clinician Working Together for Better Outcomes. Those books are available and the citations are available if you'd like to purchase them. Dr. Ahlskog was born in Chicago, Illinois, but spent the last half of his childhood or adolescence in small-town America in Cavella, Michigan. Dr. Ahlskog graduated from Michigan State University, receiving his Ph.D. from Princeton University and M.D. from Dartmouth Medical School. He interned and completed an additional year of medical residency at the University of Chicago and did his neurology residency at the Mayo Clinic. He's been at the Mayo Clinic since 1981. Dr. Ahlskog is a much sought-after speaker and an internationally known expert, and it's a pleasure to have him here today and to see him again.
Well, it certainly is a pleasure to be here. What a great city this is. You know, Cavella, Michigan. How many people have heard of Cavella, Michigan? Raise your hand. That's pretty good. It's a little. It used to be 385 people was the population, and now on the website it says about 500. So I guess they don't really know for sure, but they don't care anyway, as long as people pay their taxes. So yeah, that's about 150 miles north from here. My grandfather moved there about 100 years ago, was a farmer, and my mother was raised there. When my father died at age 11, she moved back there, and that was, for me, that was really a blessing. What a great place to grow up. I'm forever grateful for my teachers there, but I'm also grateful for the education I got at Michigan State University. And I'm from the class of 1967. That's the Bubba Smith, George Webster class, 10-10 tie with Notre Dame for the national championship. So I have a lot of fond memories about sporting events at Michigan State also. Well, we're going to talk about practical things today. I'm like a participant in Back to the Future. So we were in 2027, and now we're back to 2017. So things you can do to treat Parkinson's disease, that's the focus. And so what I'm going to be telling you are things that I believe is the appropriate way to think about Parkinson's disease treatment and how to optimize the care. Debunking 10 myths that may sabotage treatment of Parkinson's disease. I think this is a pretty sophisticated audience that is knowledgeable about Parkinson's disease, but I've just learned from talking to folks before, it's helpful to know a little bit about the background to be sure we are all on the same page. So this is the next few slides are just going to address that. You can go to sleep for 5 minutes if you like, if you know all this. So Parkinson's disease is a neurodegenerative condition, and that's an interesting concept. These are disorders where certain portions of the nervous system atrophy or degenerate, hence neurodegenerative, that term. So you recognize, I'm sure, some of the other disorders in that class. So Alzheimer's disease, you know, folks with Alzheimer's disease, unless they're very elderly, they can walk, they don't have a tremor, they're not slow, but where the degeneration occurs in the brain is especially in memory centers. So they can't remember what they had for breakfast, even with clues, but they can go out and work in the yard and have a beautiful flower garden. ALS, different disorder. It turns out that in typical ALS, there's very little degeneration. Unfortunately, it's the final common pathway from the brain, spinal cord, out to the muscles. So people that have ALS, they don't live very long because selected out for degeneration are those neurons that go to the muscles of breathing and swallowing and all the muscles, and that's what takes them away. But on the other hand, for the most part, the rest of their brain is intact. That's not entirely true, but for all intents and purposes, I think it's fair to state that. In Parkinson's disease, it's also a neurodegenerative condition. You notice these things typically tend to occur in middle age and beyond. These aren't the disorders that kids get. I mean, there are exceptions, but they're extremely rare exceptions. Parkinson's disease also is a neurodegenerative condition, and not everything degenerates. So we're going to talk about a few places in the brain, and I think this is helpful if you go online and read about Parkinson's disease, and you read about the substantia nigra and the striatum, the nigrostriatal system, and the caudate nucleus, the putamen. These are all things where I think it's helpful to have a little bit of a sense of what we're talking about here. So I highlighted a couple of terms, and the intent of the next few minutes is to get you up to speed with some of these places in the brain that you're going to be hearing about. So these are actual midbrain sections from human beings who, in the top slide, it's somebody who died and did not have Parkinson's disease, and in the bottom slide, somebody who died and did have Parkinson's disease in life. So you can see that dark area in the top slide there, that's the substantia nigra, the old neuroanatomists. You know, they didn't have electron microscopy, they didn't have stains for highlighting things in the brain, so they'd look at the brain and it was named based upon the appearance. That's why you get funny names like the inferior olive, the red nucleus, the substantia nigra, black substance. Just below, in that midbrain slice below, is a midbrain of somebody who had Parkinson's disease, and you can see the loss of pigmentation there, and that pigmentation actually marks the substantia nigral cells. So you can see now how in Parkinson's disease, this may well turn out to be an important area of degeneration, and in fact it is, but there's more to say about that. The striatum, nigrostriatal, I'm certain you've heard that term, that relates to the substantia nigra and the striatum, which are connected. So there's a pathway that starts in the substantia nigra and then goes to the striatum. The striatum is actually two nuclei, the caudate and putamen. Now, pathway, you might say, how does that work? You know, in biology, when I saw cells under the microscope, there were these sort of roundish sorts of things with a nucleus. Well, neurons, which are brain cells, which are nervous system cells, are fundamentally different in terms of their structure. So there's a nucleus, there's a cell nucleus, there are things within that nucleus that are important for the metabolism of cells, just like in all cells, but there's a long wire-like process. 100 billion neurons in the human brain, and they're interconnected. And some of these connections are rather massive, numbering in the thousands and thousands. So in the brains of people with Parkinson's disease, the substantia nigra is lost, and that connection to the striatum, hence, is lost. And that's a critical link in the movement control circuit. Those of you that are old enough and used to cut grass for $1, you know, that's what I did, and that I think a lot of guys and gals both did that. Well, the old lawnmowers, if the grass was tall, you knew you had to adjust the governor on the lawnmower. The governor adjusted the engine speed. If you're going through tall grass and the governor was set down too low, the engine would stall. So what you had to do was, if you were kind of savvy about this stuff, it was just a little wheel that was on near the engine mounting of these push gas lawnmowers. So you'd adjust the governor up. Why am I telling you this? Because that's analogous to what happens in the brain when you don't have dopamine. That governor is kind of wound down, and that governor, this nigrostriatal system, really modulates the speed and amplitude and the likelihood of movements. So you kind of know what constitutes Parkinson's disease, but if you think about it, it's like that lawnmower where the governor is set down too low. People that have Parkinson's disease motor symptoms, they move more slowly. Automatic movements like blinking are attenuated, reduced in frequency, arm swing. The amplitude of movements is lost, so you know if you go to your doctor says, do this. And people with Parkinson's disease, they may be able to do it quite rapidly, but the amplitude dampens. Just like arm swing dampens, stride length dampens. So that's the governor on the lawnmower. It's kind of wound down a little bit. And in fact, there's sort of a continuum there that relates to the neurotransmitter in the system that is listed here. It says dopamine. What in the heck is a neurotransmitter? Well, we've got this concept here of a cell body, nucleus, mitochondria, all the integrity of cells in general, a long wire-like process. At the end of that are further portions of that axon that go to different places. They're called terminals, and then they interact with other neurons. So that's a circuit. It's sort of like you know, a microchip in your computer and your television electronic sets. But the conduction down that neuron, down that axon, it is electrical until it gets to the end. But the way that the other neuron that's next in the sequence is either activated or inhibited is by release of a chemical. That's the neurotransmitter. So very few brain neurotransmitters are dopamine, But it turns out this dopamine is really crucially placed, this dopamine neurotransmitter system. So here's an actual brain slice from a human brain. So you can top his top of the head up here, and then I'm going to point to the spinal cord. And I'll talk loudly because I think you can hear me. So the spinal cord is going downstream, and so this would be right and left. And that arrow, the arrow starts in the substantia nigra, goes to the striatum, which is the caudate and putamen, and that's that crucial pathway. That's the governor in the motor control system. Cell body in the nigra, terminals in the striatum. It turns out that this is crucial to the treatment of Parkinson's disease. Arvid Carlsson, another Swede. Swedes are pretty smart folks, I guess, as we're learning today. And Arvid Carlsson won the Nobel Prize for a discovery of people's brains with Parkinson's disease were reduced in terms of their dopamine levels, markedly reduced. And that led to the discovery that you can replenish dopamine and reverse the symptoms of Parkinson's disease. Is that true for all of them? Well, it turns out it isn't true, but it's pretty dramatically effective, certainly for the things that I mentioned, the things that are like the governor and the lawnmower, slowness of movement, amplitude of movement, tremor, even though that's not that type of a problem that's attenuated, it's an extra movement, but the tremor also responds to replenishment of dopamine. So my point here is that dopamine deficiency and replenishment of dopamine is crucial to the treatment of the symptoms of Parkinson's disease, but as it says in the bottom of this slide, not everything is dopamine-based. And of course, that's what Dr. Brundin is hoping to address with all of these 2027 kinds of new strategies for addressing the progression of Parkinson's disease. That's certainly what I would like to see, because I can do pretty well treating the motor symptoms of Parkinson's disease, but there are certain things that don't respond, as shown in this slide here. This is from Heiko Brack. Heiko Brack published 2 papers, 2003 and 2004, that really crystallized our thinking about Parkinson's disease. Up to that point, the thinking about this was that this is a dopamine system. If we could only replenish dopamine, everything would be hunky-dory. We're looking for ways to give better dopamine replenishing drugs, maybe cells transplanted there. Maybe that'll answer all the questions about how we can reverse Parkinson's disease. It turns out that it isn't at all that simple, and you've already heard this from Dr. Brundin. So I'm going to talk I'm going to talk loudly again and just point out a few things. Dr. Brack, who's a neuroanatomist in Frankfurt, Germany, made the point, and this is really very consonant with the way Parkinson's disease evolves, not over months or years, but over decades. Early in life, it's a problem where it seems the pathology is in the lower brainstem. This is the brainstem. This is front. This is back. The spinal cord goes down here. The lower brainstem contains neurons that are responsible for shutting off movements when you dream. Hence, if those aren't working right, you get REM sleep behavior disorder, dream enactment behavior, and Dr. Brundin alluded to that already. It turns out that not all people with Parkinson's disease, but many act out their dreams at night. They don't know that because they're asleep and they're dreaming, but if you ask a sleep partner, oh yeah, Charlie, he's kicking and punching, and yesterday he said bad words when he was sleeping. Is that common? It's very common if you ask people about, Parkinson's disease and their spouse is sitting there, and that's the kind of response you get. The neurons that are responsible for that are in the brainstem. So Dr. Brack, based upon the review of the neuropathology, the anatomy, said that fits perfectly with what the anatomy shows us in that location, where in rats we know exactly where that location is in the brainstem, and that fits with Brack's hypothesis. The autonomic nervous system, you already heard about constipation. Now, a lot of people are constipated, and most of them aren't going to get Parkinson's disease, but if you look at 2 groups of people, somebody who's constipated early in life and another group aren't constipated, the group that was constipated later in life has a substantially greater risk of coming down with Parkinson's disease. So it's not that constipation causes Parkinson's disease, although people have argued that. Maybe it's something in the gut that's the nidus, the initiating factor. But my own guess is that probably though that alpha-synuclein process you heard about probably already is operative in the gut, and that's what's going on there to cause the constipation. And then you also heard from Dr. Brundin about loss of smell. People that have Parkinson's disease will often report that they lost their smell. Sometimes it's just recently, and sometimes it was, many years ago. And now, it's hard to know what loss of smell means because people have sinus conditions and et cetera, so you can have a poor sense of smell, and that doesn't necessarily mean that you are destined to get Parkinson's disease, but it's pretty clear if you've lost your sense of smell and you follow those people over time, they have a substantially increased risk of later coming down with Parkinson's disease. So Dr. Brock says that these are the areas where this starts. Smell loss, olfactory bulb, sits right at the base of your frontal lobes. There's the frontal lobe, there's the olfactory bulb, and the brainstem. So it turns out that fits actually with what we know about how Parkinson's disease evolves. Now you might say, what about dementia with Lewy bodies, which can be a condition that's very similar to this? The Brock scheme doesn't hold up with that, and so that's another area of uncertainty, but the Brock scheme really holds up very nicely for the evolution of Parkinson's disease. So what's my point here? Well, my point that I made in the previous slide was that dopamine loss is the substrate for treating things that are problematic in Parkinson's disease, but there are people that have an evolution, and typically the evolution occurs to at least a mild extent, where they develop problems in that blue box on the left: cognitive impairment, levodopa refractory motor symptoms. That means that you take a zillion milligrams of levodopa or dopamine agonists and it doesn't help. You still have a shuffling gait or gait freezing and so on. People that have Parkinson's disease typically get a very robust response to levodopa, but after many years, 20 years let's say, or if they reach a certain age, over the age of 80, typically you don't do as well in very aged people or people that have Parkinson's disease for long durations. And the substrate for that, I'm absolutely convinced, it just has to do with this Lewy body neurodegenerative process that has now long gone past the substantia nigra, and now it's involving other circuits in the brain. And then the other area where people get into trouble that isn't dopamine-based is the autonomic nervous system. That would be orthostatic hypotension, meaning that when you stand up, your blood pressure plummets, you feel lightheaded, you might faint. Constipation would be a component of that, but typically it's pretty treatable with a few tricks. And loss of the urinary control, so hopefully that's not a big problem. People that have Parkinson's disease, it isn't the usual things. In gals, it's having babies, and guys, it's having a big prostate. In Parkinson's disease, it's the nervous system control of the bladder that often is the reason for the urinary symptoms. So these are not dopamine-based symptoms. So I mentioned that we do pretty well as physicians replenishing dopamine and seeing some really robust and gratifying responses. This story goes back to the 1960s. I mentioned that Arvid Carlsson, he was a Ph.D. neuroscientist, so it wasn't he that led the treatment trials, but he documented that in rats you can deplete dopamine in the brain and the rat becomes Parkinsonian, and you can replenish that. The replenishment strategy that was recognized in the 1960s was to give the precursor to dopamine, that is levodopa. So the obvious question arises, well, why don't you just give dopamine? That's because there's a blood-brain barrier that protects the brain from all the different biological moieties that are circulating and if got into the brain could cause problems. So the blood-brain barrier keeps a sort of a constant molecular environment within the brain, so there aren't all the fluxes in the bloodstream from the things that you eat that go into the brain and make you psychotic after every meal. Dopamine can't cross that blood-brain barrier, but there's a transporter at the blood-brain barrier that transports the precursor to dopamine, which is levodopa. So we all have levodopa in our bodies. It's from a biologic group of compounds called amino acids. And so there's an amino acid transporter at the blood-brain barrier. So that was a huge breakthrough in the late 1960s. The FDA approved it in '69, and it was really a miraculous drug, except it took a zillion milligrams of levodopa to work, and people were vomiting. Nausea, big problem back in the 1960s. And scientists get credit for a really clever insight into this. They realized the problem was simple. It sounds simple to say now, but at the time, I think that was a pretty clever insight. They recognized the problem was the premature conversion of levodopa to dopamine in the circulation, that is outside the blood-brain barrier. So you can see that has two bad consequences. Number one, you've taken a molecule of dopamine, of levodopa, and converted it to dopamine, so now it can't get into the brain. Number two, well, it can get into the brain in one tiny area, which is the brainstem nausea and vomiting center, where the blood-brain barrier is porous. So what they did is they designed a molecule. In Europe and Great Britain, it's been Sericide. In this country, it's carbidopa. Carbidopa can't get into the brain, and it does one thing. It blocks the conversion of levodopa to dopamine. So that's been the mainstay now of Parkinson's disease treatment for over 40 years. So when I prescribe this to people, sometimes I had this lady that called me up after a couple of weeks. It kind of dawned on her. She had gone online and said, "Do you know how old this drug is?" And I did. It's over 40 years old. So she was very unhappy with me. She said, "There must be better drugs than this." But there are other drugs, but this is the crucial one. This is the natural substrate, that allows you to replenish brain dopamine. So there's a nomenclature problem here that sometimes people will say, well, I'm taking Sinemet, but these are the names for carbidopa levodopa, is what I'm talking about here. Levodopa, sometimes it's called just levodopa, sometimes it's called L-dopa. Sinemet was the old brand name for carbidopa levodopa. Those of you that went to Catholic grade school and took Latin, you recognize sine without emesis, without vomiting. So that's how Merck named that, Sinemet. And a lot of times it's still called Sinemet, even though physicians write the prescription for generic carbidopa levodopa. And now there's a new formulation of that, Rytary. Rytary is something that is used to prolong the levodopa effect. It has certain properties that I won't go into right now, but it is carbidopa levodopa as well. So you can kind of see where I'm going with this. So I'm a full-time clinician. I spend probably 60 hours a week doing clinical stuff, probably 50 hours a week with patients, and then the 10 hours that you do to do phone calls and things like that. And carbidopa levodopa, patients have taught me for years that you've got to get the dose right, and that's the best you're going to do for treating Parkinson's disease. So this talk is going to focus on how this gets screwed up. 10 myths that sabotage the treatment of Parkinson's disease. So I will read the myths, and then I'll tell you what my interpretation of these is. And these are not rare myths. I mean, these are revenue-driven myths. The competing drugs. How do you compete? If you've got a second-rate drug and you're competing with a drug that's a natural substrate for dopamine and is cheap, well, it's sort of like politics in Washington. You demonize the opposition. And so that's what happened over a period of a couple of decades. So these are the myths that people would read online or they'd read in the magazines or newspapers. Myth number one, levodopa stops working after a few years. Parkinson's disease is like aging. Every morning I wake up, I'm a day older, and I still haven't figured out how to change that. Well, Parkinson's disease is the same thing. It marches on with time. The trajectory isn't quite as, benign as aging. Aging trajectories are kind of like that, and Parkinson's disease is more like that, but it certainly doesn't stop working. Now, it is true that after 15 years of Parkinson's disease, the carbidopa levodopa is not as efficacious as it was the first 3 to 4 to 5 years, but it still works. But it's not quite as good as it was in the very beginning. And in part, that's the natural history of Parkinson's disease, and in part, it's also aging changes as well, too, that get superimposed. So if I see somebody who's, let's say, get Parkinson's disease at age 60, I'm going to start levodopa, I tell them that, well, I expect you to get within 20 to 30% of normal. That is, improved by 70, 80%. If I see somebody who's over age 80 with Parkinson's disease, de novo, now we're going to start this, I'm not quite so optimistic. So I tell them, well, I think you'll probably get 50% of normal. And so you can see here, it isn't all a dopamine story over time, and that's really the issue here. The other thing I should mention, too, is that after a period of years, which might be a few years in young people or might be 7 or 8 years in seniors, the response isn't around the clock and nice and smooth. After a while, the response gets tied to each dose. So that's called the short duration levodopa response as opposed to the long duration response. And then the timing of the doses is crucial. Early on, the timing isn't crucial except to keep the doses separate from meals. Myth number 2, almost everybody develops disabling dyskinesias who take levodopa. What are dyskinesias? Well, dyskinesias are not cramping, dyskinesias are not your toes curling, dyskinesias are not tremor, dyskinesias are wiggly movements. Those of you that used to watch The Good Wife, my wife and I used to, every Sunday evening, we'd wait for The Good Wife to come on TV, and Michael J. Fox was on there. He played a lawyer, and sometimes he was fabulous, and sometimes he was wiggly like this. Those are dyskinesias. Those are chaotic, wiggly movements. And that's driven by too much levodopa. That reflects the last dose of levodopa was excessive. So is that something we should worry about? Well, here's the incident cohort of Parkinson's disease patients in Olmsted County, Minnesota. So Olmsted County, Minnesota is where the Mayo Clinic in Rochester is located, Olmsted County. And my colleague, Dr. Van Gerpen, tabulated the people who had been on carbidopa-levodopa for 10 years, so it's quite a long time, 10 years, the frequency of dyskinesias that were sufficient to require a medication change. Now, you can always get rid of dyskinesias. You can adjust the dose. But he tabulated from the medical records, a retrospective study, what percent of patients had to have their medications adjusted because of dyskinesias. It turned out the majority did not. So 43% needed medication adjustments after 10 years, and 57% did not. More importantly, after 10 years, only 12% had dyskinesias that could not be basically controlled with medication adjustments. Now, why would that be? Well, this is sort of like that governor and the lawnmower. If it's set down too low, you can't cut the tall grass. The engine runs really slowly. And if you get the governor just right, then it runs at the right speed. If you turn the governor up too high, then it's running really fast and it's inefficient and so on. And too fast a governor is basically dyskinesias. That means you've given too much levodopa. The last dose was too high. So you go from moving too slowly to moving too much. Is it no more complicated than that? No, it's no more complicated than that. Also, it hasn't been recognized, but if you're in the clinic, because over a period of years, like I have, you realize that dyskinesias are much more common in young people with Parkinson's disease. So Michael J. Fox, apparently, according to the newspapers, came down with Parkinson's disease around age 30. So it's not at all surprising, especially after 20-some years of Parkinson's disease, that he has dyskinesias. But it turns out that if you're over the age of 70, the five-year risk, this is after 5 years of levodopa, the 5-year risk is only 16%. So that means that 84% of the people after 5 years of levodopa won't have dyskinesias. Myth number 3, also about dyskinesias. Dyskinesias are worse than Parkinsonism. Well, you have to know what dyskinesias are. They're these wiggly movements. They're chaotic movements. Sometimes it's a hand. Sometimes it's a foot that's wiggling. Sometimes it's the whole body is wiggling. Maybe the head is jerking like that. So these are chaotic movements. Note, though, that I'm using the term chaotic, so they're not rhythmic. Tremor is rhythmic. So non-tremulous movements, that's one way to recognize dyskinesias. Something that also gets confused with dyskinesias is dystonia. Dystonia is a component of Parkinson's disease in a lot of folks that have that disorder. Dystonia is a cramp-like state. Common dystonia would be toes curling. Some people, when their levodopa effect wears off, realize it's wearing off because my toes are curling. So that's a dystonia, contraction state that's reversible. That's different than hammer toes, where it's not a contraction state, it's that your tendons have shortened. But you see the point here, it's muscles that are inappropriately contracting in a sustained contraction. Foot turning in, calf cramps at night, you know, I get calf cramps as well too, but if you get calf cramps in spades and you have Parkinson's disease, it's quite possible that that is a dystonia. Tremor that's Parkinsonian tremor, dystonia that's Parkinsonian dystonia is adequately treated with levodopa. Dyskinesias are treated by reducing levodopa, and that's the point of this slide here. So this is the way dyskinesias work. It's like this scheme here shows, this is hypothetical, but I'm going to walk over here and point to a few things, and I'll talk more loudly. Dyskinesias reflect the response to your last dose of levodopa. It's not the total daily dose. So here's the schematic here. This person takes a dose of levodopa, rises in the brain, hits a plateau, and tails off. The brain has two thresholds. So one is a threshold for improvement, so you want to be above that, because a lot of the responses to levodopa are all or none. Either they work or they don't work. So you want to be above that threshold. There's another threshold that's for dyskinesias. So in a simple world, it would all be like this. You have quite a gap between the dyskinesia threshold, which is an overdose threshold, and the threshold for improvement. And so in a lot of people, you just lower each dose, not the total daily dose, and that puts you over the improvement threshold and short of the dyskinesia threshold. Now, because the neurodegeneration in the striatum, where those dopamine terminals are located, is not uniform, it's possible to have a dose that leads to improvement but always leads to dyskinesias at the same time. So this behaves like these thresholds are one on top of one another, and that happens sometimes. So Michael J. Fox probably has these thresholds that are on top of one another. So if he takes a dose, there's a very narrow therapeutic window. If he gets it just right, then he's perfect. But if it's a little bit too much, then he's dyskinetic. If it's too little, then he can't move well, has tremor, and is slow. So my guess is that as an actor, he probably needs to be a little bit overdosed, which would be a preferable state rather than underdosed. And I think that probably explains why some of the time he was overdosed on that TV series. So you can see here, this is a treatable condition. So if you were here today and he said, I'll give you $100 if you can get rid of these dyskinesias by this evening. I'm going to collect right now. Give me the $100. Lower the next dose. That's all you have to do. Or don't take the next dose, because that dyskinetic pattern will persist for maybe 2 to 4 hours, and then it's a new game again. Myth number 4, levodopa should be saved for later. And this has never made any sense to me, but this has been dogma for 20 years. Save levodopa for later. So this is like me telling my sons, well, they're now over 30, but when I used to give a talk on this subject, you know, when they were younger, I'd say, like me telling my son John, who is the athlete in our family, John, you better not play pickup basketball because you may want to play when you're my age. Some things you just can't save, and this is one of those things. And if levodopa works better 10 years now than it does 10 years from now, that has to do with the natural history of Parkinson's disease and aging than it does with something that the levodopa is inducing. So I don't think that there's any merit at all in trying to save levodopa, and as you'll infer at the end of the talk, probably you'll do better if you start levodopa when you need it. Now, you don't have to start levodopa when you don't need it. If you've got a tremor and you don't care, you don't need to start it. But on the other hand, if this is starting to compromise your life, I can't think of any reason you should save it for later, and I don't think you can do that. I think it's a lost opportunity. Corollary of that, myth number 5, the levodopa dose should be limited, and again, I think this was driven by revenue streams from competing drug companies over the last 20 years. Some of those drugs have now gone off patent, and there is not the revenue streams driving that discussion anymore. But this was what was sacrosanct in terms of how you treat Parkinson's disease, according to some people who wrote about treatment guidelines. There is no study that has looked at one group of Parkinson patients on a very low dose and another on a regular dose, except for this one that I cited here. And it was done in Great Britain. In 6 years, a group of British Parkinson's disease patients were randomized to either an arbitrarily low dose of levodopa or what they called the maximum tolerated dose, which in my clinic is the way I treat people. It's not how much can you get into them, but it's a dose that's appropriate to meet their needs. And it turns out after 6 years, the lower dose group still had dyskinesias and fluctuations, not at quite as high a frequency as the higher dose group, but they also were pretty Parkinsonian and weren't doing well. Life is short. You know, the average lifespan for males in Olmsted County, Minnesota is about 79 years. If you take big chunks of life and you say, we're going to save it for later, or we're going to be very conservative with the doses, and you don't gain anything from that, you've just lost a big chunk of your life. And I'm not in favor of that. Okay, myth number 6, carbidopa, levodopa should be taken with meals. Revenue streams haven't driven this. This has been driven by lack of knowledge about how levodopa gets into the brain, which we've known, by the way, for over 40 years as well. Levodopa is transported into the brain by a transporter molecule that sits at the blood-brain barrier. That molecule transports amino acids. So that's a class of compounds. It's actually, for levodopa, large neutral amino acids. Other large neutral amino acids, which are not levodopa, can attach to that biologic transporter. There are only so many seats on the train. So if you take carbidopa, levodopa with a meal that has protein, proteins are strings of amino acids, and when you digest proteins, you liberate all those amino acids into the bloodstream, and this is like trying to get on a train, but you only have X number of seats and you've got 12X people waiting to get on board. So that's really the problem there. There are only so many binding sites on that transporter, and if they're all occupied by other dietary amino acids, and basically levodopa can't get on board. And the levodopa response, as I mentioned, tends to be all or none. So this is why Parkinson's patients sometimes talk about failed responses. Off states that persist despite taking a full dose. That has to do with taking it with meals. So what patients have taught me, and this isn't some clinical trial that's proven this, but I can tell you that having been in the clinic for the last 36 years, I've had a zillion people who've taught me, A, you got to take it on an empty stomach, needs to be at least 1 hour before a meal to guarantee it's going to work. I realize there are occasional people that can take it 30 minutes before a meal and it works, but that's not the rule. And C, it needs to follow a meal by at least 2 hours. That's 2 hours after the end of a meal. So people that are levodopa responsive and have these short duration levodopa responses, they need to take each dose of carbidopa levodopa or bensericide levodopa at least 1 hour before meals and at least 2 hours after the end of meals. And if they do that, then they should be capturing the full effect provided the dose is right. Myth number 7, there are only 10 myths, so we're moving along. Controlled release carbidopa levodopa is a preferred formulation, and that's Cinnamate CR. Well, it was a preferred formulation about 20 years ago when it was first introduced, according to DuPont. It turns out that this is not a bad drug, but it's much more erratic. It's erratically absorbed. There's not a milligram to milligram correspondence to the regular carbidopa levodopa. I had quite a bit of experience with it early on, and what I can tell you is that it works fine in some people, but when people wouldn't get as good a response as I would like, I was never really certain if the problem was that it wasn't fully bioavailable, meaning that you weren't getting the full effect because a lot of the active levodopa was going into the colon where it can't be absorbed and hence into the toilet. So I'm not opposed to using this. I use it for certain things, but what we're going to be talking about today relates to the regular old-fashioned immediate-release carbidopa-levodopa. And I mentioned Rytary. Rytary isn't a drug that ought to be used in new patients. I mean, it would work fine, but the problem is carbidopa-levodopa costs about 10, 11 cents a tablet. This is a couple bucks or $1.50 or something like that. So my thought is, why pay all that money if the old carbidopa-levodopa works as well? So how do I use carbidopa-levodopa? Well, my colleagues and I that are down the corridor from me, we all pretty much do the same thing. So this is a strategy here. So the yellow pill there, sometimes I have to know the color of the pill because you'll ask people, what's the formulation you're taking? Is it 10-100 or 25? Well, I don't know. And then if you ask them, is the pill yellow? All these 25-100 tablets are yellow, so then I know what they're taking. Okay, you've got the pill that fits with my scheme anyway. And then what we do when we're starting this is 1 tablet 3 times a day, hour before meals, 2 hours after the end of meals. And then every week they ratchet it up, 1, 1.5, 2, 2.5. And the maximum dose I have people take is 3 tablets at a time. Why would I stop at 3? Because I've never seen anybody after 36 years who convinced me they were doing substantially better on 4 tablets at a time or 5. So there's a dosing range there. That's the dosing range that pretty much everybody fills the tank. So my usual scheme is we go through, I have a sheet of paper which looks just like this, to be quite honest. They go through that whole scheme, and I like to have them come back in about 6 weeks. Then I ask them, okay, what dose worked the best? So if they choose one, then it's a done deal. If they say, well, these 3 doses, 1.5, 2, and 2.5, all work the same. Well, common sense dictates, why take the highest if you do as well on a lower dose? So a lot of medicine is common sense, and that's one of the common sense things. And it's interesting, too, having followed a lot of folks for years, whatever we identify as the dose that fills the tank at time 0, when we first start this, typically that's a dose they're taking in 5 and 10 and 15 years. Now, they will have to take it more often, which we are going to discuss in a minute, but the dose that fills the tank is a dose that fills the tank and fills it forever, or so it seems. Myth number 8. The dopamine agonist drugs are nearly as effective as levodopa, and I'm primarily referring to pramipexole, which is Mirapex, and ropinirole, which is Requip. Nupro patch, that's also dopamine agonist. That has a different pharmacology, though, than Requip and Mirapex. But the inhibiting factor for me is it's $20 a patch. And at the maximum size patch, which is 8 milligrams, that's still subtherapeutic for the average person. It may be therapeutic for a couple of years, but this is not the long-term treatment, an 8 milligram Nupro patch. And having grown up in Calaveras, Michigan, where I still save my brown paper lunch bags, and where my wife finally broke my habit of saving all the aluminum foil, you can see I'm a little bit cheap. And if you're going to pay $20 a patch per day, you ought to cure Parkinson's disease. So you're a lot better off giving that to Dr. Brundin. Let him take that $20 a day and figure out how to cure Parkinson's disease. But point of bringing this up is that Mirapex and Requip have some very interesting side effects, and probably a lot of you have heard about the pathological behaviors. So one of the more interesting ones is pathological gambling, and I've seen a lot of these folks, and it didn't dawn on me right off the bat, but when you start to see the same story repetitively, then after a while, it kind of sinks in. People that had never gambled before, the spouse would say, Charlie can't leave the casino, and we just lost $2,000. So the operative word here is pathological behaviors, pathological compulsive behaviors. And it turns out the common theme is that anything that is inherently rewarding in about 1 person in 4, yes, about 25%, develop these pathological behaviors, things that were really getting in the way. That's how you define the word pathological. So this wasn't people that were going to the casino and losing $100. This was life-changing kinds of events. My colleague, Dr. Kevin Cullis, wrote up back in 2005, 15 men who became pathologically hypersexual. And there are all kinds of stories I could tell you, but they're not pretty, and this is an audience of Michiganders who are good church-going, synagogue-going folks, and I'm not going to embarrass you with these stories, but it was really, there were some incredible experiences that some of these people had. Pathological sleepiness occur, I don't know the frequency, but I guess it's about 1 person in 10. The first paper written on this described 9 people, 8 of whom were taking Mirapex, who got into car accidents because they fell asleep at the wheel. And with the new prolonged acting Mirapex and Requip, you can't tell that it's making you sleepy because it's long acting. You could tell a lot easier when people were on the regular Mirapex and the regular Requip because they'd take a dose and they'd say an hour later, you know, boy, I can barely stay awake. And then there are people that hallucinate. If I see a person with Parkinson's disease who's hallucinating, the first thing I'm going to do is look at their medication list. The dopamine agonists are very potent at provoking hallucinations. Carbidopa-levodopa, in my experience, either doesn't do it or does it very infrequently, which is surprising, but that's been my experience. Number 9, levodopa disrupts sleep. When I was just came on staff, I remember reading and hearing at a conference that you don't want to give levodopa at bedtime because it disrupts sleep architecture. Well, what I learned from countless patients after that is that if their levodopa effect plays out, or if they're underdosed, they cannot get comfortable in bed, and therefore natural sleep cannot occur. So there are a number of symptoms that are experienced by people with Parkinson's disease, and I think many of you would recognize this. Akathisia, feeling like they have to move but they can't move. Stiffness in bed, rigidity, can't turn over in bed, or the tremor. These are all things that inhibit natural sleep. So what patients taught me was that you have to be in a medicated state to get to sleep and to stay asleep. So if it's somebody new with Parkinson's disease, there's this round-the-clock effect with just 3 doses. So that typically would be adequate so people can sleep. After a period of some years, then this wearing off effect occurs. So if the last dose of the day is at 5 p.m. and you go to bed at 10:30, well, the effect is come and gone. Now you're lying down in bed and you can barely get into bed, you can't turn over in bed, you can't straighten out, you feel stiff, and you experience akathisia. So those are states that are incompatible with sleep. So what I learned is if people have that wearing off effect, let's say their last carbidopa-levodopa is at 5 p.m., need a dose maybe about an hour before bed. So I've done that a number of times, and I learned from patients. Somebody would come back and say, well, I tried that, but it didn't work. So I learned to ask, well, how much did you take? Well, I just took 1 tablet because I was going to bed. No. You need the dose that fills the dopamine tank because of those all or none responses. So if you at the beginning, we figured out that you need 2 tablets each dose to capture the full effect, then you need 2 tablets before bedtime. And then people would come back and say, well, that worked great, but at 4 in the morning I woke up and I couldn't get back to sleep. And then I realized the effect has worn off again. So then I tell them the simplest thing is take your full dose of carbidopa-levodopa, put it on your nightstand with a glass of water, and when you wake up, take that. It'll take an hour to work, but then you should be able to get back to sleep. Now, there's some tricks with Cinnamate CR, but that's kind of a digression. I won't go into that right now. Myth number 10. We have drugs that may slow Parkinson's disease progression, and I wish we did, but it turns out these are the biggest trials. These are 11 major trials that have some funded by pharmaceutical companies, some funded by the federal government, the National Institute of Health, the NIH, and they've all been negative. 11 drugs, 11 trials, and these are, million-dollar studies. And these are, the ones I listed here are the ones where over 100 people were enrolled. So you can see these were serious attempts to get at a drug that would slow the progression of Parkinson's disease. A lot of these were repurposed drugs, but some of these were novel drugs as well, too. So it says here, controlled drug trials were negative, no evidence for slowing Parkinson's disease progression, but. So this is what I talked about a couple of days ago to the group here, and I promised I would have a few slides that relate to evidence that regular, vigorous exercise may slow the progression of Parkinson's disease. Now, I'm a clinician in the clinic, and I'm very sensitive to all of these admonitions and recommendations that you see in TV and read in magazines. Drink 8 glasses of water every day. Take Prevacid, which is made from jellyfish that are found on the beach, and you can now take that in pills, et cetera, et cetera. So there's a lot of baloney that's out there. And so if I'm going to tell patients you have to exercise, I want to convince myself that this isn't part of that baloney litany, that's out there for all of us. So these are the arguments that convinced me that exercise is the second important component of Parkinson's disease treatment. And I'll just give you a little bit of the background about how I arrived at this. About 10 years, I read about 9 journals every month. I try to stay current. I don't read them from cover to cover, but I look at the titles and things that look interesting I read. And I started to realize about that time that quite a few articles were surfacing that suggested that aerobic type exercise, which is vigorous exercise, directly does good things to your brain. So we're not talking about preventing atherosclerosis, which it does, based upon pretty good evidence. But on the other hand, this is vigorous exercise affecting things in your brain that you would want to have affected, both to counter aging, brain aging, and also to slow the progression of Parkinson's disease. There are not a lot of Parkinson's disease studies, so this is the only Parkinson's disease slide I'm going to show you. I'm going to try to make the point, and I've just selected out a half dozen slides that I think illustrate the evidence that's out there. This paper that I just put together and submitted, it had 195 references. And I'm not telling you that to make you think that, boy, this guy writes a lot of papers and reads a lot of papers, but 6 slides, I'm doing you a favor, you're not getting all 195 slides that I could have made. So this is a paper that was published 7 years ago, and it's a meta-analysis. So it looks at a combination of studies that were all done the same way. And these are people that exercised in midlife, that ascertained the frequency of Parkinson's disease years down the road, and people that exercised compared to people that didn't exercise in midlife had a lower risk of coming down with Parkinson's disease. Now, it's not a zero risk, but it seemed to reduce that risk. And there have been 3 studies since then, one done in Finland, one done in Sweden, and one done in California that arrived at the same conclusions with each of those studies. Well, if I could change one thing in the future, if I could prevent some component of Parkinson's disease, I would choose dementia. There's a 6-fold risk of dementia, but it's not 100% risk. But when you develop Parkinson's disease, there is some risk of mild cognitive impairment. There is a risk of more substantial cognitive impairment. And the idea would be, if I had my druthers, is something that would reduce that risk substantially. So there are no clinical trials that have ever been done that have looked at exercise or anything in long-term risk of dementia. So we're going to have to look at the data from normal folks. So these are people this is another meta-analysis, and this is, again, 11 different studies, people that exercised in midlife versus those that didn't. They combined the data, and the outcome measure they looked at here was dementia risk. And it turns out that people that exercised in midlife, I already mentioned exercise folks in midlife have a lower risk of Parkinson's disease, and the same is true for dementia. If you exercise in midlife, those that exercised had a lower risk of later coming down with dementia. This was published in 2009. There have been 4 studies since then. They all showed the same outcomes, except one of the 4 was not statistically significant. The other 3 were. So it would suggest that if you exercise in midlife, you should have an association of lower risk of Parkinson's disease, lower risk of dementia. And if we're worried about dementia as an outcome of Parkinson's disease, I think that's relevant to that. But there are different ways of explaining data as well, too. Maybe people that were destined to become Parkinsonian or demented, maybe they also were more apathetic and less inclined to exercise as well, too. So we have to look at some other parameters. How about brain volumes? As a neurologist, I look at a lot of MRI brain scans. And the first thing I do before I start paying much attention to the scan is I look at the age. If the person is over age 80, I'm expecting kind of a walnut kind of brain, because that's unfortunately what happens as you age. Your brain shrinks. Not good. And is that uniform? Yeah, it happens to everybody to variable extent. Some brains shrink less than others, though. So given a choice, you want to choose the less shrunken brain. And if you think about it, this process is already starting earlier in life. You know, if you read about the great mathematicians who have proven Fermat's theorem, whatever that is, these papers are written in the 20s. The mathematicians are on the downslide of their career, you know, once they're in their 40s or in their 30s. People that are athletes who have good joints and haven't had orthopedic injuries, baseball players, can't turn on a fastball at age 40 anymore. Well, that's just reaction time, but reaction time is mediated at the brain level. So a lot of things happen as you go through life, and there's a natural aging process that occurs, and the brain shrinks. So there have been a number of studies that looked at brain volumes. The MRI scans are basically, this is data that can be interpreted, modified, and subjected to software programs. So clever people can use software packages, and the one that's usually used here is this software called FreeSurfer, where you can look at the volumes of brain. So one area that's been looked at is the hippocampus. The hippocampus, as you probably know, it's a memory area. You can see in that slide up on top is this hippocampus, the two yellow areas, that's the location of the hippocampi. If we had a monkey and lesioned their hippocampi, the monkey would have no short-term memory. So this is an area that's crucial for short-term memory. So given a choice, check off the box that says bigger hippocampi. So there have been two studies that have been done. Erickson's group at the University of Pittsburgh. I'm going to talk more loudly and point. This is fitness. This is least fit. You can measure fitness. How do you do that? Well, you get somebody on a treadmill or an exercise bicycle. You have somebody there cracking a whip. They exercise the maximum amount. Then you look at the volume of oxygen uptake. Physiologists have used this for decades to document degree of fitness. So that's called VO2. Poorly fit, very fit. So if somebody were running 5 miles 3 times a week, they'd be somewhere on this end of that curve. Okay, this is left hippocampus and right hippocampus. This is the size of the hippocampus. It says volume. The volume of the hippocampus is what I would choose for my hippocampus. I like a big hippocampus, so my memory is good. At least that's what I would presume. And you can see there's a linear relationship between how fit you were and the size of your hippocampi. And that, I just happened to run across that. I thought, wow, that's really pretty interesting, and I think it's relevant to a lot of us in this room and outside the room. Then Erickson did a prospective study, that same group, University of Pittsburgh. He took a bunch of seniors and randomized them to a stretching and toning group. So this is where you're kind of doing stuff like this, you know. That's the control group. And then a vigorous exercise group, and as I recall, it was cycling. They would cycle 3 times a week for an hour, and they would have somebody there that would be, kind of encouraging them so people didn't get lazy. And then they measured the size of the hippocampus at baseline in 6 months and 12 months. And interestingly, the hippocampus got larger, that's the blue line up on top, after 6 months of exercise, and even larger yet after 12 months. Now these are small changes, so these aren't people whose hippocampus is growing out of their ears, but it's measurable things on an MRI brain scan. But you can see the red line, that's the group that was doing all this stretching kinds of stuff, and I mean, that's good. I'm not opposed to that, but it doesn't have the effect that you saw in the group that's illustrated by the blue line. So vigorous exercise was associated over a year's worth of those endeavors with a larger hippocampus, and if you were just not doing that and just stretching and doing the usual stuff, your hippocampus probably, it's an age-related phenomenon, the hippocampus was shrinking. And this has been replicated in other studies shown in that box as well. Cortical volume, so the cortex, that's the covering of the brain, and that brain that I showed you, there's the midbrain, and then there's the small area that's kind of in the middle, and there's a thick cortex, and this is where a lot of stuff is encoded. If you can shoot a free throw, that's where it's all encoded up there. If you can do complex math, that's the part of your brain that you use. If you're a good writer, the writing codes are up there. The dictionary in your mind, that's up in the cortex. So you want to have a big cortex also. So check off the box, big cortex. That's what you're going to choose. So it turns out here, I just illustrated a number of studies. Now, there aren't any negative studies in all the data that I've shown you. So you might say, well, if it's a negative study, if they didn't find that, they wouldn't publish it. And that's probably true. There's something called publication bias. But as I was reading this literature, I realized that, boy, there are very few studies where people found the opposite, where exercise made their brain smaller or didn't change anything at all. So what I'm showing you, it's not selected just for the positive studies. It's selected for the studies as representative of what I was finding in that literature. So you can see here it says cross-sectional studies of fitness. People that were fit had less brain cortical atrophy over at a point in time. So people were assessed for the degree of fitness, that VO2 max measure that I mentioned that physiologists used, and then their MRI scan cortex volume was looked at. The more fit you were, the less brain atrophy, the larger cortex you had. And there were prospective studies, 3 of which were positive, 1 was negative, that showed that 6 months of vigorous exercise was associated with a larger cortex, slightly larger, but statistically significantly larger, and only 1 negative study, which I've indicated there. And there are 2 studies shown below where people reported physical activity. So one of those, they reported it, and then they looked at the white matter volumes, and they were larger in the people that reported more vigorous physical activity. And then in that last study, it's another Erickson study from the University of Pittsburgh, walking distances, the longer you walked, then they had their cortex measured as well as cognition measured 9 years later, and the walking distances at time 0 were associated with larger cortices. And this is, I think, the last slide in this component of exercise I'm going to show you, the Baltimore longitudinal study of aging. And this was funded by the National Institute of Aging, and it was implemented by the National Institute of Aging and Johns Hopkins. And this is years of life, so age. And these were people that were enrolled in Baltimore, and they underwent a fitness test, that VO2 max that I mentioned, how fit they were. So I'm going to walk to the slide and I'm going to point here. These are 4 different cognitive measures, components of an IQ test. So on this score here, the greater the number there, the more errors you made. And there are 3 trajectories here. This is words learned. The solid line are the least fit. The dotted line are the most fit. So for each of those measures, fitness was only measured once, but the assumption was if you were fit at time 0, you had a greater likelihood of being fit throughout the whole study, which lasted 18 years. The more fit you were at time 0, the more favorable your trajectory on these cognitive studies. I lied. I do have a couple more slides. Animals. You can do things to animals you can't do to humans, like cut off their heads and measure things in their brain. That's why the animal studies are so interesting, because, I mentioned that it looks like exercise directly does good things to your brain, which you can measure in rats. So if you go back to this slide here, put a running wheel in a rat's cage and they'll run like crazy, or put them on a treadmill and force them to run. Things that you would want going on in your brain, BDNF, what does that stand for? That's like putting fertilizer on your lawn, brain-derived neurotrophic factors. So that's something that's measurable. And 27 out of 27 studies, animals that were exercised elevated the BDNF in their brains. 3 studies looked at GDNF, that also went up. And those of you may remember that about 8-10 years ago, there were 5 different studies that looked at people with Parkinson's disease having cannulas implanted into their brains to infuse GDNF, thinking that that may help slow the progression of Parkinson's disease. Maybe exercise would have done that. Neuroplasticity. Those of you who are leaving and going out to your car today, you have to remember where you parked your car. That's mediated through neuroplasticity. So the brain is constantly reprogramming itself and remembering where you parked your car, what your hotel room number was, etc., or remembering something you read in the newspaper. This is all mediated by neuroplasticity. So these brain neurons are forming new connections all the time, and neuroplasticity is what you want going on, because if that weren't working, we wouldn't be able to remember anything, and we wouldn't be able to do very many things. So it's an ongoing active process, and all the parameters of neuroplasticity I've published here. There are no negative studies. Neuroplasticity genes are expressed in greater volumes in animals that are exercised. Synaptic plasticity proteins, such as funny names like synapsin or synaptophysin, relevant to the synapses and so on. Neurogenesis. Neurogenesis relates to the hippocampal cells, one of the few areas in the brain where you can make new cells. Neurogenesis goes up in the brains of animals that were exercised, hippocampal neurogenesis. Long-term potentiation. Long-term potentiation is the neurophysiologic substrate of memory. That is enhanced by exercise, as are dendrite complexity, which is on the other side of synapses. And one point here, and then I'm going to close with the last slide. Levodopa became available in 1969. 7 different medical centers published on longevity rates just before it became available and just after. And 7 out of 7 studies documented substantially increased longevity with the advent of levodopa. What is going on there? Well, you could argue that, well, maybe levodopa has something to do with a neuroprotective effect per se. I think we just liberated a whole generation of Parkinson's disease patients who are confined to wheelchairs and beds in nursing homes. Final slide. 2 simple rules for treating Parkinson's disease. Number 1, I think you need to optimize levodopa when the time is right. If you're doing great and you don't care, you don't need to start it. But if your life is starting to become curtailed by the limitations, then we want you as good as we can get you, and then once you can do that, then engage in vigorous exercise. We have time for questions?
I think we need to move on. What I was hoping is that we could take about a 5-minute exercise break to stop the progression of our dementia. During that break, if people have questions for Dr. Ahlskog, they can come down here and ask them, and then we need to restart in about 5 or 10 minutes, okay? All right, could we come back in and take a seat? And we need to keep moving because we have lots more interesting information to disseminate. All right, it's my pleasure now to introduce Dr. Christos Sideropoulos, who's an assistant professor in the Department of Neurology and Ophthalmology and the Movement Disorder Clinic at Michigan State University. Dr. Sideropoulos is a board-certified neurologist with subspecialty training in movement disorders, including Parkinson's disease. He is a graduate of the Aristotle University Medical School in Thessaloniki, Greece, and subsequently received psychiatry residency training at the University of Erlangen, Germany. Interesting, I don't know if you all realize, but it's the American Board of Psychiatry and Neurology, and there's a lot of interchange between psychiatry and neurology. And it isn't unusual for people to go back and forth from one to the other. He completed his neurology residency training at Henry Ford Hospital, as well as his fellowship training, and pursued further specialization in deep brain stimulation at the University of Toronto, which is really one of the premier centers for working with deep brain stimulation. Dr. Sideropoulos has published over 25 peer-reviewed papers, co-authored several books, book chapters, served as a reviewer for scientific journals, and is involved in a number of clinical trials. He's going to be talking today about non-motor symptoms of Parkinson's disease, which I think is often a neglected area of treatment that is something that's very important for patients. So this is a very interesting presentation. Thank you.
Thank you very much, Dr. Cullis. All right. Good morning, everybody. Thank you very much for the honor and pleasure of inviting me here. I would like to congratulate Dr. Cullis, Mary Sue Lanigan, and the Van Andel Institute for organizing this, which is a wonderful event and hugely important for everybody. I have no disclosures about the presentations I'm going to give. I would like to say that the non-motor symptom story is a recent story, a recent recognition of how neglected this aspect of Parkinson's disease is. It is important to recognize, and it has been stated already, that throughout the clinical course, patients with Parkinson's disease exhibit both motor and non-motor issues. However, as it was stated before, well before motor symptoms happen, and later on, once essentially patients enter into a kind of a burned out phase, non-motor symptoms may predominate the clinical picture. As a side note here, I would like to mention, not to make this very exhausting to everybody, that there is a big overlap with Barbara Fisher's presentations when she talks about the psychiatric aspects of Parkinson's disease. So I'm going to keep that part of my presentation relatively brief, so not to be quite burdensome. As mentioned, the non-motor symptomatology up until recently has been hugely under-recognized and underreported. It does, however, impose a huge burden on families and caregivers. And it is important to state here that part of those symptoms can be to some degree responsive to levodopa. So the notion that levodopa cures everything is wrong, but also on the other hand, the notion that levodopa does nothing about the non-motor symptoms is wrong as well. And Dr. Ahlskog also mentioned that, for instance, levodopa can improve quality of sleep, which is a big truth, clinical truth in our everyday practice. So it is important that we recognize that we screen our patients for non-motor symptoms, and to the degree possible, we treat them. And in my discussion, I consider guidelines from the American Academy of Neurology, the European Academy of Neurology, and the Movement Disorder Society. However, there are not that many large, well-controlled, well-designed studies, and a big chunk of this is clinical practice and wisdom that we get from our patients. So in talking about non-motor symptoms, it is nice, in order to keep ourselves relatively organized, to lump them into big categories. So the first one I chose as the category of sensory dysfunction. The second one has to do with mood disorders, and it is closely related to cognitive disorders, and part of it is going to be mentioned briefly because Barbara Fisher is going to talk to you more about that. Sleep disorders are a major player that reduces quality of life and can happen many, many years before the manifestation of motor symptoms. We're going to talk about autonomic nervous system dysfunction and briefly about speech issues. And there are also some other non-motor symptoms that are not very easily classifiable, but we're going to just mention them just to understand how broad the spectrum of non-motor issues in Parkinson's disease is. So in terms of sensory dysfunction, it has been already mentioned that a substantial portion of Parkinson patients, not all of them, some studies mention that there is sensory dysfunction up to 90% of Parkinson patients, cannot smell very well. And this may happen many years before Parkinson's disease occurs. And why is that? We don't know for sure, but we postulate because one of the first sites in our brain, our nervous system, where the Parkinson's disease pathology starts is the olfactory bulb. That's the center of smell, so to speak. Typically, that problem does not get better with levodopa treatment, and we need to recognize that. And perhaps other neurotransmitters may be implicated, for instance, acetylcholine, which is a chemical that mediates memory. But when you go to your doctor, the doctor needs actually to ask about this question. Oftentimes, we ask patients that we suspect they may have Parkinson's disease. They say, you know what, I have problems with sense of smell. perhaps constipation, sleep problems, then you start thinking, aha, this makes sense. This may be early Parkinson's disease. It is always interesting to try to exclude reversible causes like a big flu or some cold, some trauma that has damaged the olfactory bulb, vitamin deficiencies, and so forth. But in itself, that problem may not be reversible. So going on and moving forward to other sensory dysfunctions, visual hallucinations are also commonly associated with Parkinson's disease, typically later on. Earlier on, we're talking about a cousin of Parkinson's disease, namely Lewy body dementia. They're associated with cognitive issues and sleep dysfunction, and oftentimes they're actually worsened by dopamine treatment, levodopa treatment. Another problem that is not very well recognized is the impaired color vision and low visual acuity. Oftentimes, people with Parkinson's disease, they will say either they see double, which may have to do with slowness of the eye globe moving muscles, but they oftentimes perceive colors in a different way, and they report that they can't focus very, very well. It is postulated that this may have to do with degeneration, with dying of nerve cells in the retina, in the back portion of the eye. And you may not be aware that many of those cells use dopamine as a chemical, neurotransmitter. The important thing to understand about Parkinson's disease is this is a whole body disease. It's not like a brain disease. It is a skin disease, a gut disease, a salivary gland disease. So whatever you have in your body cells, nerve cells, and especially dopamine-producing cells, you may see some issues there. Another very well established but quite underreported, under-recognized, and under-treated problem is pain in Parkinson's disease. And pain may have a plethora of causes, lots of different causes in Parkinson's disease. To some degree, it is associated with Parkinson's disease itself, because the basal ganglia, where the problem typically not happened, but it's probably focused during most of the Parkinson's disease course, they also process sensory input. That means the lack of dopamine has also to do with how our body processes painful stimulation, so sensory inputs. But it may also be related to off symptoms. As medications wear off, pain can be part of the disease. It can also be related to painful off dystonia, to musculoskeletal deformities. So lots of factors that we need to consider carefully when we evaluate our patients. An easy thing to do, again, as Dr. Ahlskog mentioned, is to optimize levodopa. And I cannot say how much I agree with what has been said, that oftentimes you see patients who fluctuate, who wear off frequently, and you start seeing them like shaky, stiff, and frozen, and they report also pain gets worse. So make sure this is optimized. The other interesting thing is that in Parkinson's disease, not only dopamine, but lots of other chemicals get disrupted, including serotonin and noradrenaline. And those chemicals, among others, have to do with pain perception. So as I mentioned, when we see patients with Parkinson's disease, make sure we optimize their dopamine treatment, reduce the off time, which is oftentimes associated with painful perceptions. Sometimes using extended release levodopa may help, especially during sleep. And again, Dr. Ahlskog mentioned that, and this makes perfect sense. Oftentimes, patients are stiff and frozen in bed. They can't fall asleep. You give them like an extended release levodopa, which again, as was mentioned, is not optimal, but may give them something throughout the whole night, and then they say, aha, I sleep better. Levodopa or apomorphine pumps may also help sometimes in terms of pain, although they are not actually approved to treat mostly non-motor symptoms. Those other invasive approaches, also including another known player here, deep brain stimulation, they may, apart from improving motor problems, improve also non-motor problems, including pain. So some patients, as we all know, they develop painful dystonia, either the toes curling down, especially early in the morning, or sometimes the big toe, especially going up, something we call striatal toe. In those cases, again, you try to optimize medication management, but that might be quite annoying, quite painful, and may we have to resort to botulinum toxin injections. Sometimes in clinic, we think combining Okay, some non-steroidal anti-inflammatories or antidepressants may help, and physical therapy can also be helpful sometimes. So moving on to another domain is mood disorders. Anxiety disorders are also very common. You'll see every time I mention a domain, it is extremely common. That means Parkinson's disease is the epitome of a neuropsychiatric disease, a movement, but also mood, but also cognitive, but also sensory disease. Up to 60% of Parkinson's disease patients have been reported to have some sort of anxiety problem, which is sometimes, not always, associated with depression. And anxiety can be a very early manifestation of Parkinson's disease in the preclinical or early stages. It seems to be more common in women, but is also very common as the disease progresses, oftentimes in conjunction with depression, which can be oftentimes to some degree reactive when patients are not properly treated. The anxiety can be a part of what we say non-motor fluctuation. That means as the medication wears off, anxiety kicks in. The same applies to depression. It can be improved with dopaminergic therapy, including the standard Sinemet, but also dopamine agonists, and sometimes deep brain stimulation or other treatments, invasive treatments like the pumps, may help. But again, don't think that, okay, medications may not work so well, let's go ahead with DBS to treat anxiety. This is just as a side note. Antidepressants sometimes are prescribed, preferably the newer serotonin selective reuptake inhibitors, the newer modern antidepressants to treat anxiety, and as we'll see later, depression. And they may help sometimes, and it is important to think about using them properly. So as mentioned, depression, clinically significant in up to a third of Parkinson's disease patients. It is typically milder than the depression seen in non-Parkinson patients. It is less severe and not that commonly associated with psychosis, such as seen in psychotic depression in non-PD patients, but it's more commonly associated with another hugely under-recognized and under-treated issue, apathy. Apathy means lack of motivation, lack of energy to do stuff. So the depression can be due to the disease itself, or reactive, or separate, or all of the above. And many neurotransmitter systems can be involved in depression, including dopamine, noradrenaline, and serotonin. That's why oftentimes we use antidepressants that may affect multiple neurotransmitters. In those cases, a multidisciplinary approach is necessary, involving not only the neurologist who can recognize what's going on, but also a psychotherapist and a psychiatrist and a counselor. The problem is, in real life, finding that multidisciplinary approach is really difficult and extremely challenging and frustrating for us as clinicians. The treatment can be some sort of newer antidepressants, mentioned in brief here as serotonin reuptake inhibitors or serotonin and noradrenaline, like Cymbalta, the duloxetine, like Celexa here, for instance, and tricyclics. Now, tricyclics are the older-fashioned medications, and it is nice to consider, perhaps, avoiding at least tricyclics like amitriptyline, which causes also worsening of memory. Now, there was a recent publication from this institute, actually, very recent publications, mentioned that a tricyclic called nortriptyline may actually slow down the clumping of alpha-synuclein, and then you get those questions. I mean, should everybody be getting, instead of the newer ones, nortriptyline, which, again, I would probably consider doing that in the context of a clinical trial, not just randomly, because every time you see a patient, you have to factor in the whole profile of the patient. It is important to also know that dopamine agonists like pramipexole and ropinirole have an antidepressant effect in Parkinson's disease. This is something that is not very well recognized. But again, dopamine agonists, as Dr. Ahlskog mentioned, those are tricky medications, not straightforward. You have to think about potential side effects. Electroconvulsive therapy has been discussed and has also been reported to improve Parkinsonian symptoms, except for Parkinsonian depressions, repetitive transcranial magnetic stimulation, cognitive therapy, and so forth. So multiple potential modalities, and they have to be employed within a multidisciplinary approach. Deep brain stimulation, keep in mind, in some patients may exacerbate depression. So whenever you see a patient who already has depression and may be a candidate for deep brain stimulation, you need to carefully consider whether this is the right choice for the patient. Apathy, it was already mentioned. Up to 60% of patients with Parkinson's disease, with or without depression, sometimes the patient is just apathetic, but they may look depressed, and may or not coexist with dementia. Can be an early manifestation of Parkinson's disease out of proportion to the movement issues. A patient can be moving okay, but you see, the spouse complains, he's doing nothing. He wants to watch TV all the time. And unless properly recognized, it becomes a chronic, untreated problem with other sequelae, for instance, with reduced activity. Apathy can also be a side effect of deep brain stimulation. This is important to recognize. Sometimes you can have both depression and apathy as early side effects of deep brain stimulation. Dopamine is involved in apathy as well as acetylcholine. That's why sometimes optimizing dopamine treatment and perhaps using, in some patients, medications for memory, which enhance acetylcholine, the memory neurotransmitter, may help. The treatment, as we mentioned, is proper recognition, dopamine agonists, cholinesterase inhibitors, the memory medications like Aricept or donepezil. All that is within the clinician's experience. There are no great trials out there to support this. Sometimes we even consider using amphetamines to treat apathy. And as you see, there are no great, very well-designed trials, but this is essentially wisdom that we have gained from our patients. Fatigue, another big issue. Up to 50% of the Parkinson population, troublesome to treat, may have multiple causes, not only because of Parkinson's disease itself. It is not associated with the duration of severity of disease. Now, fatigue can be due to Parkinson's disease. It could be because of depression, which is common in Parkinson's disease. It could be due to sleep disturbance. Think about obstructive sleep apnea, especially if they mention, my husband or wife snores a lot. Could be perhaps also because of low blood pressure. It is very common in later stages of Parkinson's disease to see patients who develop blood pressure drops, and fatigue can be a major player in this, so a big problem. Sometimes, keep in mind, fatigue can be a side effect of levodopa, a rare side effect. Not so rare, but it is relatively uncommon. People say, I take Sinemet. Every time I take Sinemet, I fall asleep. Dopamine agonists can cause sleepiness and fatigue. In those cases, you may have to lower the individual doses. There's no great treatment for fatigue induced by levodopa. In some cases, you can try some coffee. Okay, amphetamines. There is some evidence to support the use of amphetamines. Amantadine, which is an old-fashioned medication initially used to treat the flu, but also used in Parkinson's disease quite frequently, and modafinil to also tackle some daytime sleepiness as well. So mood disorders, impulse control disorder, and OCD. This can be either because of the Parkinson's disease itself or as a side effect of the dopamine agonists. That's why dopamine agonists are useful medications, but also tricky ones. It's a relatively common side effect, impulse control disorders and compulsive behavior, perhaps up to 50% or even more in patients who take that. That's why it's important to counsel our patients and tell them, if you want to try this medication, we decide this may be a choice, be aware of that. When you deal with those issues, as I mentioned, sometimes it's part just of Parkinson's disease. It's also important to discuss stopping any offending agents, especially when we're dealing with impulse control disorder like gambling, hypersexual behavior. Important to think about discontinuing slowly and carefully the dopamine agonists. Now, deep brain stimulation as an invasive modality may help reduce dopamine agonist dose and indirectly help impulse control disorder. So keep that in mind. However, the interesting thing is sometimes impulse control issues can happen as a side effect of deep brain stimulation. So the important take-home message here is invasive therapies like DBS oftentimes can make things better, but sometimes they can have some side effects, and you need to weigh pros and cons when you recommend them to patients. There is some limited evidence that amantadine may help with obsessive-compulsive behavior in Parkinson's disease. And neuroleptics. Now, neuroleptics is probably not a so great choice. Neuroleptics are medications used for schizophrenia. And especially the classic ones are definitely not a good option. I would say no option in Parkinson's disease patients. I have seen up until recently some patients who have been placed on newer generation neuroleptics, which have less of a potential to worsen Parkinson's disease. This may be kind of like a second or third line measure if everything else fails. No, pseudobulbar affect. I mean, it is, again, a relatively under-recognized issue in Parkinson's disease. That means an inappropriate, emotionally not congruent laughter or crying. This also happens very commonly in the geriatric population, in Alzheimer's disease, for instance, in patients with Lou Gehrig's disease, in patients with stroke, but it also happens in patients with Parkinson's disease. So there are no guidelines about how to deal with this problem. You can consider a medication that has been approved to treat pseudobulbar affect in the geriatric population called Nuedexta in those cases, because pseudobulbar affect can affect a substantial proportion of Parkinson patients. Now, keep in mind that part of the medication may be an issue in patients with heart disease. And again, you can consider the same drug for patients with agitation, following the paradigm of patients with Alzheimer's disease and agitation. So, cognitive issues. As the disease progresses, people with Parkinson's disease, they start getting more and more some cognitive difficulties. I don't use the word dementia. Dementia means that the memory issues are so severe as to impair activities of daily living. But after 20 years of Parkinson's disease, the vast majority of Parkinson patients, they have some sort of cognitive issues, including Parkinson's disease-related dementia. So the degree of Parkinson's disease-related dementia seems to correlate with the Lewy bodies, those nasty things in the cortex, in the outer part of the brain, which controls cognition and memory, along with the hippocampus. Now, what can we do? Drugs. Rivastigmine is the most well-studied, the so-called Exelon, including the patch, the Exelon patch, which we commonly use in daily practice. But also other medications, similar, very similar ones that are used in Alzheimer's disease, like Aricept, generic being donepezil, or galantamine. The brand name is Razadine, can be used in Parkinson's disease-related dementia. They seem to have a moderate at best effect. They may help, they may work for some patients, but clearly they are not a good treatment for all patients. Some patients, for instance, can get more confusion. If that doesn't work or the result over time wears off, someone may consider adding memantine or Namenda, which is used as a combination treatment in advanced Parkinson's disease, advanced Alzheimer's disease. Or you may want to elect memantine if one of those drugs is contraindicated, because those medications can lower heart rate, for instance, may have cardiac side effects. The benefit varies, keep that in mind as we mentioned that. The interesting thing here is in Parkinson's disease dementia, and even more so in dementia with Lewy bodies, where visual hallucinations can be an issue, those medications may be probably the first thing to use, not only to improve memory, but also to reduce hallucinations. That's an important thing to consider. And as an off-label thing, we'll talk about that later, is using pimavanserin in patients with Parkinson's disease-related dementia and psychosis. And I'm going to mention that medication in just a brief moment. Pimavanserin was just approved a few years ago as the first and only medication to treat Parkinson's disease-related psychosis. That means Parkinson's disease that is associated with persistent hallucinations and or delusions, false beliefs, and seems to have a much better side effect profile compared to the typical and atypical neuroleptic medications. So sleep disorders. Sleep and wakefulness is commonly disrupted in Parkinson's disease, very common. And as it was mentioned multiple times, if you see a patient with REM, sleep behavior disorder, like thrashing around and yelling in their sleep and punching their wives or something, and yelling, and you follow those patients up for many years, it's almost certain they're going to develop some sort of neurodegenerative disorder, either Lewy body disease or Parkinson's disease, or a Parkinson's disease look-alike, which is multiple system atrophy. So those are the so-called synucleinopathies. That means neurodegenerative conditions where Lewy bodies get accumulated in the brain, and they are characterized by REM, sleep behavior disorder. So the sleep disorders not only include REM, sleep behavior disorder, but also insomnia, difficulty, not so much falling asleep in Parkinson's disease, but mostly staying asleep. Also, excessive daytime sleepiness can be a problem in Parkinson's disease, oftentimes associated with obstructive sleep apnea, periodic limb movements in sleep, like somebody moving their legs and without necessarily having vivid dreams, and also circadian disruption. That means a reversal of sleep-wake cycle. So typically we sleep at night, we're up during the day. Oftentimes the opposite happens in Parkinson's disease patients, and you will see that also sleep hygiene is quite poor, because patients with Parkinson's disease, what happens is they get insomnia, they don't sleep well at night, they take daytime naps, they get rested during the day, and they're up at night. So sleep disorders are quite, I would say, extremely common in Parkinson's disease, and they do significantly affect quality of life, mood as well, and cognitive performance, because if somebody doesn't sleep well, if somebody has untreated sleep apnea, then what happens? There is a substantially increased risk for memory loss, substantially increased risk for memory loss. So, as we mentioned, insomnia and sleep fragmentation is very common, up to 80% of Parkinson's patients. It may be due to the Parkinson's disease process itself, because Parkinson's disease also affects centers in the medulla, in the center of the brain, that have to do with regulation of the sleep-wake cycle. But it can also be oftentimes a side effect of medications, including Parkinson medications. It could be sometimes because of psychiatric issues arising in Parkinson's disease. Keep in mind, depression can also manifest as too much or not enough sleep. So better sleep hygiene, as I mentioned before, is really important, especially avoid daytime napping if at all possible. Increasing physical activity and light exposure is another modality that is not very much used, but it can improve sleep hygiene. Now, what do we typically do in clinical practice? Benzodiazepines, like for instance clonazepam, can be used, but the problem is this may worsen confusion and memory. Quetiapine is typically used, and doxepine. Those are medications commonly used in clinical practice. Are they optimal? Most likely not, because they also affect memory. A nice thing that I personally and many other clinicians use probably as a first-line thing is melatonin. Melatonin is probably a low-risk, low-benefit medication. Use it properly. That means usually use higher doses, not 1 milligram or 2 milligrams, perhaps start with 3 or 5 and go up to 10, perhaps even more. It's a natural sleep hormone. Your own body produces melatonin. Take it for a couple of weeks to see if there is any benefit. Extended release levodopa may improve sleep. DBS may improve sleep. The Duopa, the Cinemet pump, may improve sleep, although they were not approved or intended for that purpose. Again, RBD mentioned it is also a risk factor for cognitive decline. Important to note. Patients with more severe Parkinson's disease, they're more likely to have a REM sleep behavior disorder. So it may cause safety issues. Maybe patients can fall off the bed. Medications that we use, of course, after aggravating medications that are like antidepressants, clonazepam, as we mentioned, melatonin, first-line medications. Interesting here, there was a small trial using the Exelon patch, or rivastigmine, which mentioned some benefit in terms of improving REM sleep behavior disorder. And dopamine agonists may help, but they are not consistently beneficial here. RLS, quite common in Parkinson's disease, but it is not the same thing. So that doesn't mean everybody who has RLS will get Parkinson's disease. Associated, not always, sometimes with low iron levels. Dopamine agonists are first-line treatment for restless leg syndrome, but keep in mind, dopamine agonists can cause impulse control issues, so counsel your patients appropriately. There was a recent study a few years ago about using Lyrica, a well-known neuropathy drug, that showed actually that Lyrica worked as well and had less of side effects. One of the side effects of treatment is augmentation. That means you get symptoms earlier during the day and you need to go up on the dose. Avoid dopamine blocking medications, replenish iron if needed. It's always nice to check iron levels. And sometimes you may actually, instead of using the dopamine agonists, I have changed my practice. I typically like gabapentin and pregabalin to treat restless leg syndrome as a first-line thing instead of putting patients on dopamine agonists. Now, the interesting thing is deep brain stimulation, again, can help. In some patients, either less. In some patients, can make it worse. Sleep disorders, briefly, periodic limb movement is in sleep. Levodopa may help, may reduce fidgetiness of the legs. Sleep disorder breathing, mainly sleep apnea, weight loss, think about CPAP, referral to a sleep clinic. And circadian disruption, think about light therapy, but again, you have to find a physician who knows how to use that therapy appropriately. Typically psychiatrists or some sleep physicians are trained to do that. We mentioned briefly about excessive daytime sleepiness, multifactorial assess for sleep apnea. Modafinil is the classic medication that we use, which is a stimulant. You can try some extra coffee. Methylphenidate is one potential option. Amphetamines, another antidepressant called atomoxetine, and of course counsel against driving because there is an increased risk of car accidents. Now, the interesting thing here as a side note, new evidence suggests that people who fall asleep deeply, they progress more slowly in Parkinson's disease. So one thought here would be, okay, if we promote actually deeper sleep without REM sleep behavior disorder and thrashing around, could we potentially slow down Parkinson's disease? This is just a side note, an interesting, intriguing thing. This has not directly to do with doing something today, but perhaps future trials could go that route. Now, another big problem, autonomic nervous system dysfunction in Parkinson's disease. Urinary, bladder problems, erectile dysfunction, GI issues, heart and blood pressure issues, and some other interesting things that are worth mentioning. Now, urinary dysfunction, it was already mentioned. If you exclude, for instance, like bladder issues, prostate issues, or if you exclude OBGYN issues in women that have had multiple childbirths, think about other possibilities in patients with Parkinson's disease. So it seems that the pathology affects some inhibitory control of the brain to the bladder. That means instead of the brain taming and reducing, bladder contractions, the opposite happens in Parkinson's disease. Medications, sometimes medications like L-Dopa or dopamine agonists can help a little bit. Up until recently, very common medications like oxybutynin have been used to treat overactive bladder in Parkinson's disease. However, the vast majority of those medications, they have adverse memory side effects. That means they worsen memory. A favorite medication that I personally prefer is this one, mirabegron, which is a novel class of medications, beta-3 agonists, that doesn't have cognitive side effects, and in some patients may have a good side effect, which is increasing blood pressure, especially in those who actually drop their blood pressure. They have orthostatic hypotension. What else can we do? Intranasal desmopressin to actually thicken actually urine. Sometimes we have seen improvement in bladder capacity and voiding after subthalamic nucleus DBS, Botox injections, some more weird neuromodulation approaches, which are uncommonly employed, are neurostimulation of the sacral nerve or the posterior tibial nerve, but again, you'll have to find the right neurosurgeon to do that. Those are uncommon neuromodulation approaches that seem to, however, work. Erectile dysfunction. It does happen in Parkinson's disease. Keep in mind, when we see a patient in clinic with early Parkinsonian features, like a male patient with early Parkinsonism without diabetes, and the patient mentions early on erectile dysfunction, that may be a red flag for a Parkinson's disease lookalike. So we always think about that, but it does happen in Parkinson's disease. Usually after several years, Viagra is classically used, but you have to make sure the patient doesn't have any heart issues, because it can lower blood pressure, especially in patients who take other blood pressure medications. So another very common thing, GI dysfunction from top to bottom. So we start with classic thing, drooling, especially as the disease progresses. Drooling means excessive pooling of saliva in the mouth. The interesting thing is, in Parkinson's disease, typically there is not excessive saliva production. There is actually less saliva production, but because patients with Parkinson's disease swallow more slowly, all secretions get pulled in the mouth. Dysphagia, difficulty swallowing, impaired gastric emptying, and constipation. Now, as I mentioned, drooling, very common in Parkinson's disease patients, especially in late stages, and it is associated with an increased risk of respiratory infection. It may coexist with dry mouth. As I mentioned, saliva production is less in Parkinson's disease. Dry mouth is also present in Parkinson's disease, and sometimes also caused by medications for Parkinson's disease. And the whole problem is aesthetically disturbing, but also bad for oral hygiene, especially dryness of mouth. Now, drooling, what can we do? Sugarless hard candy or gum. Simple measure. It doesn't always work. It doesn't always help patients. In that case, we can try a medication called glycopyrrolate, which helps dry secretions. At least in theory, it may have less of cognitive side effects, because it lowers a chemical used for memory in the brain, but it may cause constipation. Atropine drops that are used, for instance, for asthma, underneath the tongue can be used. Scopolamine patch, but again, you can have similar side effects like constipation, adverse memory effects. And botulinum toxin injections, typically in the parotid glands, here at the angle of the jaw, and or very uncommonly done in the submandibular glands. So dysphagia, difficulty swallowing, choking, quite common, especially as the disease progresses. Dysphagia, however, can also happen relatively early in the disease, and unless you ask the patient and the caregiver multiple times, you may not pick this up. It is subjectively reported in a third of PD patients, but if you do swallow evaluation studies, it can be as high as 82%. Aspiration, that means food goes the wrong way, can be present in 15 to 56% of PD patients. And that can be a big risk factor for pneumonia, aspiration pneumonia, especially in institutionalized patients. Speech and language evaluation is clearly necessary and early on. You don't have to wait. In reality, almost every Parkinson patient, with the exception of the very mild ones, would need eventually a speech and swallowing evaluation and treatment. A rehabilitative approaches, diet modification may help. Sometimes an evaluation by a GI specialist. There are some sort of modalities, some sort of exercises that can be employed by a speech pathologist, such as expiratory muscle strength training and video assisting swallowing therapy. Those are things that typically the speech language pathologists who deal with Parkinson patients can employ to help our patients. Impaired gastric emptying and GI motility. In 70% to 100% of Parkinson patients, it may result in delayed or complete loss of benefit from levodopa. The interesting thing is this guy here, H. pylori, the bug that is associated with stomach ulcer, may play some role here by affecting inflammation and or gastric, like stomach motility, and it may be implicated in fluctuations of motor symptoms by impairing the absorption of levodopa. Another thing that has been reported in Parkinson's disease patients is the so-called small intestinal bacterial overgrowth symptom. That means too much bacteria in your gut, and this may also have to do with motor and non-motor fluctuations. So there are no really good studies what to do about impaired GI motility. remove or optimize exacerbating factors. We can use prokinetics, like medications that make the whole GI system move a little bit more. Don't use Reglan. Don't use Reglan or metoclopramide. Use domperidone, which is not that cheap. Oftentimes patients get it from Canada. Cisapride has had some cardiac issues. Erythromycin is an antibiotic that makes your gut move a little bit faster. Consider eradication of bugs with antibiotics. Sometimes orally disintegrating levodopa like Parcopa using non-oral routes may tackle the issue of impaired GI motility and inconsistent benefit from medications, or DBS or apomorphine, which can be injected. So, a big problem, constipation. Early on as a premotor issue in Parkinson's disease, but also throughout the whole disease. It can substantially affect quality of life. It is probably the most common GI symptom in Parkinson's disease. It is reported, if you carefully ask patients, in the vast majority of Parkinson's patients. But it can also be a side effect of Parkinson's disease medications like levodopa. So levodopa can cause constipation. And also patients who have duopa pump, the cinnamon pump, may report constipation, as well as dopamine agonists. So what do we do? First and above all, drink water. I bet 90% of Parkinson's patients don't drink enough water. Most of the time, especially in men, because they're afraid of going to the restroom too often. If you don't drink enough water, nothing will happen. So you're going to remain constipated forever. Okay. So increase, the fear of going to the toilet makes you constipated. So you may want to choose, about toilet versus constipation. Increase diet fiber, fruits and vegetables. You can use fiber supplements like psyllium metamucil, increase physical activity, really important, and avoid prolonged periods without any bowel movements. If the stool stays in the gut for like a week, then it becomes like cement. Nothing can move it, okay? Miralax. You can use Miralax. Add Miralax to your oatmeal, perhaps. It helps retain water in the GI tract. You can perhaps use lactulose syrup and other osmotic laxatives that help, again, pull some water in the gut. Probably a last-ditch resort, if nothing else works, is a medication called Linzess, which actually increase the secretions of the gut and makes the stool a little bit softer and easy to be pushed out. So cardiovascular symptoms. Very common in Parkinson's disease, especially a big problem is orthostatic hypotension. And it can be a nasty problem, difficult to deal with, and usually goes hand in hand, I would say, with cognitive dysfunction. Sometimes you can also see labile hypertension. That means the patient has a roller coaster blood pressure between too low and too high. There are several issues in Parkinson's disease patients, such as increased heart rate at baseline, and also a reduced maximum heart rate during exercise. Now, it is a difficult problem to deal with. Recently, we got another newly approved drug by FDA called droxidopa, also known as Northera, which can be titrated more slowly. It has been in Japan for many years. The other FDA-approved drug for neurogenic orthostatic hypotension is midodrine. Watch out for supine hypertension, so you don't take it and lay flat. You have to be upright for 3 to 4 hours. Other medications commonly used are fludrocortisone, which helps actually retain water, and a medication used for myasthenia gravis called pyridostigmine. Sometimes we use them in combination. Again, drink water. If you don't drink water, nothing will happen. You have to fill your tank with water. And the other thing is, we have to rationalize the existing blood pressure management, so it doesn't make sense to take amlodipine 10 milligrams and lisinopril 40 milligrams, and then complain about orthostatic hypotension. So you need to discuss this with your doctor, and also think whether your Parkinson medications may lower your blood pressure. Sometimes too much Sinemet can lower blood pressure. So this is important to rationalize, and at every visit, discuss with your neurologist, your medications, and with your family doctor as well. So some other stuff, interesting, that are not very often discussed. Polyneuropathy, it is not that uncommon in Parkinson's disease patients. That means some sort of diffuse nerve damage. Mostly sensory, that means your feet cannot feel well enough. sometimes associated with vitamin deficiencies, okay? Because Parkinson's disease patients, because they have GI issues and poor mobility, oftentimes they end up malnourished. So vitamin deficiencies are extremely common in Parkinson's disease patients. The neuropathy sometimes can be exacerbated by the L-DOPA or in patients with DOPA pump. Now, whether Parkinson's disease itself causes polyneuropathy, this remains an issue of debate. Oftentimes, we check B12, B6, and folic acid levels in the clinic. If they are low, we replenish. If there is too much B6, like in multivitamins, we keep an eye on that and we stop supplementation. Diabetes is also common in the geriatric population. Another interesting thing is a runny nose. Okay? Up to a third of patients with Parkinson's disease may have a runny nose, and this is also under-recognized. It's not just because they have allergies. It is not very well associated with disease duration, more common in men. Sometimes you use atropine spray to dry up your mouth, your secretions, the same spray that we use to dry mouth secretions. Dysfunctional sweating sometimes, especially as a non-motor manifestation as medications wear off. It's a poorly understood phenomenon. Sometimes you can have like dry hands and very sweaty body. Sometimes dysfunctional sweating can be a side effect of deep brain stimulation. So you see, those are interesting things that are not very well recognized. We oftentimes neglect to check our patients. So speech issues, very common speech issues, something we call hypokinetic dysarthria, both dopaminergic and non-dopaminergic pathways involved. It is worth treating patients with enough Cinemet to make sure you get rid of whatever stiffness and slowness of the laryngeal muscles. You can get rid of it, and then whatever is left is non-dopaminergic problem, like something that doesn't get better with Cinemet. The highest level of evidence suggests benefit from the classic speech therapy for Parkinson's disease called LSVT, Lee Silverman Voice Training Loud, with improvements lasting up to 2 years. Now, there are some other approaches. You may have heard about something called Vital Stim, which is using surface electrodes to kind of give a massage to the vocal cord muscles. This needs to be validated. There is no great evidence supporting this. And finishing and hopefully leaving a moment or two for questions. Very valuable resources in terms of getting information. I have, again, to congratulate the job done by the Michigan Parkinson Foundation, are the Davis Finney Foundation, Michael J. Fox, our beloved Michigan Parkinson Foundation, and the NPF as well, including lots of educational materials. And with that, I would like to thank you for staying awake. Okay.